Good morning, everybody. It's great to be here and to be welcomed by Bill. I would like to thank the organizers for including me in this exciting program. I look forward to share some of our recent work with you and start by introducing some of the general concepts in the field of chromosome folding. So the question that brings us all together here today and this week is the question is, how do cells fold their genomes? And it's more than just asking how are chromosomes folded, although we want to know how that works. We are particularly interested in understanding which molecular machines fold chromosomes and through which mechanisms. And ultimately, of course, we want to understand how chromosome folding relates to gene expression, DNA replication, chromosome inheritance, DNA repair, all these genome functions that we care about. If we think about mechanisms of chromosome folding, there are, in reality, just a handful of mechanisms and principles that we know of that fold chromosomes in vertebrates. The first one is, oops, too many buttons on this thing. The first one is compartmentalization, which was actually one of the first phenomena seen in both the microscope and in high C later, which is the spatial segregation of heterochromatin, euchromatin, and centromeres, for instance. Here's a beautiful picture taken by the lab of Irina Solovay. So spatial compartmentalization of euchromatin and heterochromatin is widely known. The phenomenon is well known, but it's actually kind of surprising that the mechanisms that drive this and the biophysical processes and the biochemical phenomena, including the actors, the proteins and so on, that mediate such segregation are still, I think, one of the least understood principles of chromosome folding. We know currently much more about things like chromatin looping, a phenomenon that has also been seen over many years, including by biochemical experiments, imaging experiments, and later by chromosome conformation capture. And I will talk a lot about chromosome looping and the machines that do this in different situations. A third phenomenon that gets much less attention, but actually should get a lot more attention, is topological features of chromatin. This is originally a very classical way of thinking about chromosomes, including in bacteria and circular molecules. But it's a phenomenon that's not getting, I think, sufficient attention in our field. We often think that in vertebrates, we don't have to worry about it so much because we think, to a large extent, chromosomes do not entangle with each other very much or internally. But recently, we showed in collaboration with Leonard Murney's lab, that actually sometimes chromosomes can be topologically entangled internally. Loops can become catenated, chromatin domains can become entangled. Today I will talk not necessarily about any of these three phenomena, although I introduced the first two in more detail about what we know about them. I will really focus on a new idea that is emerging, that in order to fold chromosomes in different ways, either during different stages of the cell cycle or in different organisms, you really do not need any additional mechanisms. You can run these same phenomena, these same principles in different ways by regulating them in different ways. And that gives rise to chromosome folding states that can look dramatically different. So in order to understand chromosome folding, we not only have to understand how each of these principles work, but their interplay is really key, and how any of these mechanisms are regulated are going to turn out to be really rich sources of areas of study in the future. And that's what I will focus on today. So again, I will not really talk about topological features, but I think in the future, this will be a growth area in our field. Okay, so if you think about interphase chromosomes, I want to go over those first two principles, chromosome compartmentalization and loop formation, in a little bit more detail about what we know about them and how they contribute to chromosome folding. So as I said, in the microscope, you can see a folded genome here. And you can see in red here heterochromatin at the periphery, in green euchromatin in the center. Here in blue we have chromocenters. It's obvious that chromosomes are compartmentalized in three-dimensional space. This was known for many years, and in high C we recognized it very early on. Here's a high C map of human chromosome 14, and you have this beautiful checkerboard. It's one of the first features we saw almost 15 years ago. And we now have a deep understanding as to how this checkerboard of long-range interactions emerges. And the reason the checkerboard emerges is that along the linear chromosome, chromosomes are composed of a series of domains, here in red and blue, and these are the classical heterochromatin and euchromatic domains. And in three-dimensional space, heterochromatic domains, for instance in red, I always mix up the colors, actually blue is heterochromatin in this slide, interact with other blue domains but not with red domains, and the red domains interact with red domains but not with blue domains. And that gives rise to this checkerboard, but what it represents in three-dimensional space is that somehow heterochromatic domains cluster together, euchromatic domains cluster together, and this is readily detectable. And again, this is one of these phenomena that we know, all of us know about this. We are very familiar with this. It's known for decades, maybe longer. But how is this formed? And I think this is still something we don't quite know, although phase separation is an obvious candidate by a physical process that will drive these associations or repulsions between opposite types of domains. Many people, I probably missed people, and I apologize, many people in this room and in the field have contributed ideas and models to explain this separation. And I think the major question going forward is really like, what are the molecules that determine these affinities for each other, and how is it actually regulated? One thing that has emerged over the last several years is that this is actually quite a simplistic view of chromosomes, where there are just two types of chromatin. We now know from the work from Ernst Aiden's lab a number of years ago, later followed up by some work by us, led by George Spraklin and Nazar Abdener, to show that there's more than just two types of chromatin that leads to two phases in a way, if you think about phase separation. In reality, and this is really was spurred by much deeper high C maps, giving rise to much more detailed structural insights, combined with much deeper epigenome maps. And we realized that, in fact, instead of just having A and B, or these two types of chromatin compartments, it turns out you can split that in several types of active chromatin and several types of inactive chromatin. Each of these have unique long-range interaction patterns in high C. They have unique chromatin modification states seen by epigenome mapping. One thing that I find particularly appealing in this study that we published a few years ago is that, yes, in this particular cell line, HCT116, we can identify these 5 different types of chromatin domains and 5 different long-range cis and trans interaction patterns. But these are not universal. Some of these don't exist in other cell types, and some cell types may have additional profile patterns that are not identified in this cell type. So there's much more diversity and much greater richness to this type of chromatin feature. And I think going forward, it would be really interesting to see how these domains change their shape, their characteristics. For instance, during differentiation, this B0 domain, for instance, is really appealing for a transition state between different types of heterochromatin. So again, putting this in context of regulation and development would be a really interesting future direction. So again, as I said in the beginning, that we don't know much more except the phenomenology of compartmentalization. We know much more about chromatin looping because we have actually molecular mechanisms. And we have molecular machines that make loops, and that is always very appealing for experimentalists because now you can perturb the system, control the system, and work with and study the system in atomic detail. So over the years, as we started to analyze these high C map at much higher resolution, and now, of course, increasingly, many people are using micro-C or region capture micro-C to really get nucleosome resolution contact maps. We started to appreciate that at much smaller scales than this checkerboard pattern of compartmentalization, we can see at much smaller scales of, say, tens to hundreds of kilobases, a great diversity of structures that are seen in high C that we originally called topologically associating domains. We now know there's much more complexity than just domains. In fact, we can see yes, there are domain boundaries, and of course these are often bound by CTCF, but there are loops which are point-to-point interactions in these contact maps, and there are line-like features. And initially when you look at this, you would think, oh, these are lots of different structures. I will probably need lots of different mechanisms to explain them. And it turns out you only need one mechanism. Okay? And this is the common theme in this presentation today. You can make wildly different chromosomes or conformations of structures with just a single process that is implemented and regulated in specific ways. So now we can identify looping interactions, and in fact, work by many people in this field, many of them are here in the audience. We now know the machines that make these loops. And those are cohesins and condensins, and we'll hear a lot about it at this meeting. Some great exciting talks I look forward to hearing later today. And I'm sure in the last several days these proteins were mentioned a lot. I just show here cohesin and condensin. There's also an SMC 5-6 complex that also can make loops, but its role in chromosome folding is currently less known. So these cohesins and condensins look very similar. They're these ring-shaped complexes, multi-subunits, and for a long time it wasn't quite clear what they were doing, but we now know these are ATP-dependent loop extruding machines. So that actually was all started with an experiment done several years ago by Kees Dekker's group, where he could show by single molecule experiments that if you add DNA on a slide and you add this condensing complex, it can make loops in an ATP-dependent process. Here's my single molecule experiment. So the idea is that these complexes can bind DNA and start pumping out loops. And that process itself, that's basically the engine that drives chromosome folding. And what cells do is use this engine in all kinds of different ways to fold chromosomes in all kinds of different shapes, and that is what I will go over today. Turns out if you just assume it makes loops and that's the only thing it does, this is a simulation from Leonid Murney's lab a number of years ago. If you have, for instance, a cohesin complex that can extrude loops, it can start at any place along this chromosome, but it gets blocked. It gets blocked by it gets blocked by CTCF. You can get an ensemble of conformations. An individual snapshot of such a conformation doesn't look too informative, but if you do a simulated high C experiment on an ensemble like this, you get features that look very much like what we see in high C. Okay, so this is a simple process of loop extrusion with boundaries is sufficient to explain all these complicated structures I just mentioned, including domains that we used to call topological association domains, but also boundaries and lines and dot-like structures and loops and so on. So it's really appealing that this simple process can give rise to all these complicated structures. And this led us a few years ago to come up with a rather simple, maybe in retrospect naive or even wrong, model that says, okay, these domains made by loop extrusion blocked by CTCF sites may play a role in gene regulation. And the idea at the time was that maybe enhancers can regulate target genes within this domain, but given that this locus can never be extruded all the way to this other gene because of the boundary in between, such enhancer wouldn't regulate this gene. It's a very simplistic model in a way, and there's a lot of genetic evidence in support of this. There are many cases where such boundaries get deleted, exposing a gene to an inappropriate enhancer regulating the gene in the wrong condition. But there's also good evidence that this is not the right model. You can remove cohesin that makes these loops, and it has surprisingly little effects on gene expression. But the good thing about models is they lead all of us to come up with new questions that we can ask. And I think I just listed here a couple of questions that I think are really big questions in our field currently that we might need to think about trying to answer. And I'll give you a few things we have done to try to answer these questions. The first is, what cis elements control cohesin loading? We actually know surprisingly little about this. In the simulation I just showed you, there is no preferred loading at any specific site. We'll hear more about cohesin loading at enhancers later from a wonderful talk from Leonid Murney's lab. How is it loaded? Biochemically. A lot of structural work is being done on questions like that. What determines extrusion speed? Actually, what is the extrusion speed in vivo? And I have something to say about that later in the talk. What determines processivity? Is it a highly processive process, or does it stop and go? What DNA elements control stopping or pausing or unloading of the cohesin or any other extruder? How does it get unloaded? what is the molecular mechanism of extrusion? And maybe I'm just not very good at structural biology. When I look at all these beautiful cryo-EM structures of the cohesin complex, I still have a hard, or condensate, I still have a hard time understanding how extrusion actually works. So I find it very difficult to have a picture of how this motor actually does its job. And then, of course, it's not just a canonical structure of a complex. I'll say a little bit about that in a second. What is the subunit composition and the conformation of cohesin during loading or extrusion or blocking or unloading of this complex? And I know we have some exciting talk coming up about PDS5, one of these subunits of cohesin that I think has a very interesting role. And of course, the key question in the end is, does it play any role in gene regulation? And maybe it does, maybe it doesn't. Maybe the function of cohesin really is to fold chromosomes. Okay, so here are some outstanding questions, and I'll give you some examples of the work we're doing to address some of these. So first is a story that was led by Anne-Laure Falton. She's now a group leader in Toulouse, and Sergey, a senior scientist in my group, who explored this simple idea of a topologically associated domain with random loading of cohesin blocked by CTCF sites and possibly bringing enhancers and genes together. I'll just summarize some work from us, but also some other groups, and I apologize again if I miss a few references here, because again, there are so many contributors to this field. One thing we proposed based on all work from Anne-Laure and Sergey was that there is a preferred loading of cohesin at enhancers. It starts extruding, and it maybe stop or pause for a while at promoters. Once it's inside the body of an active gene, it gets pushed out, and I did some work on that area too. Once it's the 3' end of a gene, it gets rapidly unloaded by WAPL. So that we call this a cohesin traffic pattern, where cohesin is actually running around on chromosomes, but actually doesn't always reach the CTCF sites, even though that's where we see a lot of the loops occurring. We also see loops between enhancers and promoters. What I find fascinating about this is, beyond the fact that there are specific cis-regulatory elements that control any of the steps of the extrusion cycle, all the elements that do that are involved in gene expression. So all these type of cis elements that people have been studying for years, turn out each of them seem to have some kind of a control function for the loop extrusion cycle. Suggesting, of course, a direct link with gene expression, or maybe not, we don't know yet. But this illustrates a much more deeper complexity than these original models we and others had proposed of random loading and blocking at CTCF sites. This whole process is regulated precisely by all these different cis-regulatory elements. Second, what about the cohesin complex itself? We like to draw it as this ring-like structure that can embrace two pieces of DNA. Originally it was found as the complex that can hold sister chromatids together, but does it do loop extrusion in the same way? So we were inspired by a beautiful experiment several years ago from the Wendt lab, where they tested this idea of cohesin being a ring and topologically embracing DNA, and whether that ring formation was essential for chromosome folding. And the idea was really implemented, their idea was implemented in another system by Sunny Liu, a postdoc in the lab at the time. He now leads his own lab at Fox Chase. And the idea is very simple. And again, this is really from Kristen Wendt's paper, the idea. If cohesin rings embrace DNA, then cleaving the ring with a protease should dissolve all loops in vivo. Okay, so here we have another way of looking at a topologically associated domain. Cohesin is found, of course, at the boundaries holding the CTCF sites together, but there's actively extruding cohesin complexes in the middle of the domain. So what Sunny did, he introduced a protease cleavage site in RAD21, one of the subunits of cohesin. Cleaving RAD21 in that way is sufficient to separate sister chromatids. Okay, so we know by doing so, the ring gets opened. We know that from work from Kim Naismith and many others, that when cohesin holds sister chromatids together, it does really topologically embrace two pieces of DNA. Opening the ring is sufficient to let that go. That interaction is gone. So Sunny did the following experiments, where we take G1 nuclei, expressing this RAD21 that can be cleaved by a TIF protease, add the protease to cleave all cohesin, wait a little bit, and do high C, and ask which loops stay and which loops are gone. And it turns out you can cleave all cohesin It's all no longer an intact ring. The only interaction you lose is those between the CTCF sites. You do not lose cohesin loops inside the domain. Okay? Actually, those cohesins can be washed off with salt. So that led Sunny to propose that there's actually two types of cohesin, one interphase chromatin. There is cohesin in the middle of the loops, actively extruding. It holds the DNA not as a ring, topologically embracing two pieces of DNA, even though we often draw loop extrusion that way. Inside these domains, it's somehow clamped, a new conformation also seen in cryo-EM. It's clamped tight on the DNA, but it doesn't topologically embrace it. However, by the time this cohesin is at the CTCF site, and particularly when it holds two CTCF sites, cohesin now, something happened, something mysterious happens, and now this complex changes either its shape, subunit composition. Now it's a ring that has opened and probably has now embraced two pieces of DNA. So it's a very different cohesin complex. Maybe that's why this is a long-lived complex. Some of these loops can live for quite a long time. So, and in fact, Jan-Michael Peters' lab has shown in a completely different way also that the complex that mediates CTCF interactions is very related to the complex that holds sister chromatids together, very similar to our conclusion here. And we were excited that we could exchange these results before publication with them to make sure that we gave each other credit. Okay. There we go. So, and in fact, this makes a lot of sense with some structural biology. As I said, this clamped conformation, which has been seen by cryo-EM, by Shi et al. Several years ago, is probably the actively extruding complex in the middle of the topologically associated domain, but it's a different complex than when it holds the CF sites together. So this is an example of how we went from understanding very simple ideas about loop extrusion and domain formation and loop formation to a much more sophisticated view of this process, where now we know on the DNA side all these cis elements that control the process, the loading, the stopping, the unloading, the blocking, it's all regulated by these cis regulatory elements. On the protein side, we know this complex is not just this universal ring-like structure. It has probably different conformations when it loads, when it extrudes, when it pauses, and when it unloads, or even gets stalled at CTCF sites. It's probably a different conformation and may have different subunit compositions. So this really illustrates that besides the fundamental process of loop extrusion itself, the basic mechanism, how it actually runs in cells, which elements control it, which subunits get involved, actually determine how the chromosomes fold. So it's the regulation of the machine, not the machine itself, that I think we should be looking at going forward. So, how are we doing? Doing good. As I said in the beginning, there are really three known phenomena. I briefly introduced compartmentalization and loop formation and topological features that I won't go into much detail of. But I highlighted the fact that it's all about controlling these processes if you want to fold chromosomes or change the folding of chromosomes. It's a differential control and interplay between these mechanisms. Can explain a wide array of different conformations. For instance, during the cell cycle. So we talked mostly about interphase up to now, and we have these beautiful contact maps. But when cells go into mitosis, we heard, of course, Bill's beautiful keynote lecture yesterday. We get the most beautiful chromosome, the mitotic chromosome. And cells can go from an interphase state into a mitotic state in literally 10, 15 minutes. It's a remarkably fast process. And by high C, the chromosome conformation also changes quite dramatically. How does that work? When we first started looking at the cell cycle dynamics of chromosome folding, you would imagine that to make this structure or this structure, you might need very different mechanisms. And again, the theme of my talk today is that you don't need different mechanisms. Those same few, small number of mechanisms is sufficient to explain this structure and this structure. The difference is how these mechanisms are implemented and regulated. So we heard a lot from Bill yesterday about the organization of mitotic chromosomes and how chromosomes are folded, the role of scaffold proteins and condensins. And I want to talk a little bit more about how this chromosome is actually acquired by the actions of loop extrusion. few years ago, also with Leonid and Bill's lab, in collaboration, we showed, through many Zoom and Skype calls, that the mitotic chromosome is an array of loops, which of course have been known from Uli Lemley's lab and Bill's early work, and some early work from us and others. But we finessed that model and found that in fact there are two types of condensins that make mitotic chromosomes. Here in red we have condensin 1, makes big loops, and we found that it gets broken up in smaller loops by condensin 1. So we have nested loops, loops within loops, made by the two condensins. And second, we found that there's some intrinsic helical path of these loops inside mitotic chromosomes. This phenomenon is how that it arises is still unclear. And one of the points is, again, what I want to make today is that in order to make this structure, you don't need to come up with a new mechanism. Again, it's loop extrusion. But what happens is that in interphase, chromosomes are made by cohesin-mediated loops, whereas in metaphase, something happens and it gets replaced by condensin-mediated loops. So there's basically an exchange of the machine that makes the loops. Also, there's other things happening that I won't talk about a lot. Compartmentalization is turned on in interphase and somehow gets turned off in mitosis, at least in part. And again, we have recently shown that most of the time chromosomes are entanglement-free, but in mitosis, chromosomes become topologically entangled within each chromatid. So what I want to focus on today mostly, actually, is looking at what happens during, let's go back one slide. But what happens during this transition, because it provides a beautiful example of how just implementing loop extrusion in different ways and how machines that make loops interact with each other to go from interphase to metaphase, focusing mostly on this switch from cohesin to condensin-mediated loops. And again, looking at this transition from interphase to metaphase, which happens really, really quickly, gives us an opportunity to study the interplay between cohesin and condensin and how they regulate each other or interact with each other during this transition. Now this is an important moment to indicate a really special case of going into mitosis. And that is that during the entry into mitosis, there are at least four different SMC complexes on the chromosome. We're not mentioning SMC 5, 6, we haven't found a big role for that. There are at least 4 of these SMC complexes acting simultaneously on the chromosome. We have extrusive cohesin, which is still there because we're in interphase. At the beginning, we're exiting G2. We have loops made by extrusive cohesin. We're in G2, we have sister chromatids. They're held together by cohesive cohesin. And then, as soon as we go into into mitosis, the condensins get activated, initially condensin 2 and later condensin 1. So we have 4 highly related complexes on the chromosome. Okay, so they're all acting simultaneously. Now, I won't have time to go into any details, but we were able to estimate the extrusion speed of, for instance, condensins. In vivo, it's been measured in vitro on naked DNA, but in vivo, the extrusion speed is between 1 and 3 kilobase per second. Okay, that's on chromatin, inside cells. And I still have this mental picture in my head that I find rather extraordinary, that I'm thinking about these long chromosomes, and we have these machines, right now, these machines run along the chromosome, 1 to 3 kilobase per second. It's a busy place. These complexes are running around. What happens when they run into each other? They're gonna run into each other. How is such a conflict resolved? And that's really what we're gonna explore going forward. Okay, so I wasn't quite honest when I showed this picture of a mitotic chromosome. This is just one sister chromatid with the nested loops and the spiraling loops. We have, of course, a sister chromatid. So there's a second copy of this structure. They're held together by these cohesive cohesins somewhere in between the two sister chromatids. And by the time you form this structure in prometaphase, the extrusive cohesin that make loops in interphase has somehow been evicted or has left the chromosome. How do cells go from interphase within 10 minutes to build this structure? And that's what we're going to try to explain. Okay, so again, as Bill pointed out yesterday, it's really important, and it was actually a key technological breakthrough for this project, is to have the ability to enter mitosis in a highly synchronized manner, in a very efficient manner, so that we can do not just imaging, but bulk assays with high-C to follow chromosome conformation as cell populations synchronously go through mitosis. So again, it was introduced yesterday. This was a cell line made by Kumiko, where CDK1, which is the major kinase driving cells into mitosis, is mutated so that it can be inhibited by NMPP1. So that NMPP1 can be washed out, and then the whole cell population will just march through mitosis in a remarkably synchronous way in these chicken cells. So this is the cell system we're using to look in real time, at minute time resolution, how interphase structures get converted into mitotic chromosomes. That combined with cell lines in which we can deplete using an oxygen-inducible Dekker system that I'm sure many of you are familiar with, that Kumiko made a remarkable panel of cell lines where we can deplete either both condensins or individual condensins or cohesins or all the combinations thereof. So we can isolate, we can study chromosome formation by the wild type set of complexes or in situations where we have only one or different combinations of SMC complexes running on the chromosome. So here we have a cytological view of what happens when cells go from G2 into mitosis. I think this is a 30-minute time point. So you can easily see that in normal cells, G2 cells interphase nucleus, they form mitotic chromosomes within a reasonable amount of time. If you deplete condensins, that doesn't happen, as Bill showed yesterday. The chromatin does compact or condense, but you don't form these rod-shaped chromosomes. Here's the high-Z view of the same timeline, where we have, I split the contact maps in two. So on the bottom is just a control cell. We have a beautiful checkerboard pattern, the compartmentalization I talked about early on. If these cells go into mitosis, after 10 minutes, that structure is erased. And then you start forming the mitotic chromosome with its beautiful second diagonal that Bill talked about yesterday as well. Now, interestingly, if you deplete condensin, that doesn't happen, and even at late time points, we can still see evidence of compartmentalization. And, in fact, loop formation by cohesin, as I'll show you in the next slide. So clearly, condensin is somehow actively erasing the interphase structure. How does that work? So here we zoom in a little bit, looking at loops made by cohesin. Here is a CTCF loop in an aggregate in G2 cells. If you deplete if you go into mitosis after about 10 minutes, those cohesin-mediated loops have been erased. But in the absence of condensin, when we add auxin in G2, we remove condensin and go into mitosis, those cohesin-mediated structures remain. We can quantify that. You can see at G2 we have set at 1 a level of compartments, or we call it dots, which are loops. They go away over time in control cells, but in the absence of condensin, that happens, but much slower. Okay, can we model this? Can we understand how this works? And this is really beautiful work from Samir in Leonid Murney's lab. And this is the idea behind the simulations. This is actually a simulated data set where we have loops in G2. They're made by this green complex, which is cohesin. And as cells enter mitosis, condensin comes and also starts making loops. So now we have two loop extruders on the chromosome. Condensin extrudes loops. At some point, it's going to run into this cohesin. What will happen? Samir simulated 4 different scenarios. It could just unload the cohesin and just keeps going. It could not unload cohesin, but just as a little bulldozer, push it in front of it. Or it just ignores it, steps over it, and starts making more complicated loop structures. Or it just runs into it, stops, and can't go any further. So what happens in simulation is that in order for these cohesin-mediated structures to disappear over time, only the unloading and pushing models will work. They get weaker over time, whereas the loops stay very strong when you allow bypassing or stalling. Here's another way of looking at that in a somewhat different view. If condensin can unload or push cohesin, you lose all these features typically associated with cohesin. If they bypass or stall, nothing really happens. So we think extrusive cohesin is at least probably unloaded. We can't really separate unloading and pushing, but we have some proteomics data from Bill's lab and other ideas that we like to think it can unload cohesin. So that brings us to our first rule of engagement. Our first rule of engagement for SMC complexes is when condensin encounters an extrusive cohesin, it will remove it. Okay. A second feature of mitotic chromosomes that I introduced earlier on is that if you look at a pair of sister chromatids in prometaphase, here's one pair of beautiful imaging from Komiko. In red, we see condensin located in the core of each of these sister chromatids, but in between the sister chromatids is cohesin. This is cohesive cohesin, not extruding cohesin. This is cohesive cohesin that holds the sister chromatids together. How did it end up there? If you take a whole bunch of images and you just take cross-sections and average that over a number of chromosomes, you can see that in blue we have the DAPI. It's hard to see the two sisters as two masses. They're pretty squished together, probably by cohesive cohesin. Condensins are in the core of each sister, but cohesin is not co-localized with condensin. It is in between the sisters. Again, we use simulations. Again, this is Samir's work. To test different scenarios as to how cohesin could end up there. So now we start simulations with two sister chromatids, held together by cohesive cohesin in green, and we enter mitosis, and condensin comes in and starts making loops in cis along each sister chromatid. What happens when this condensin runs into a cohesive cohesin complex? In this case, we have two scenarios. The first is it will just step over this cohesive cohesin complex and keeps extruding. As a result, cohesin ends up at the tip of the loops. Or condensin doesn't know what to do. It gets stuck on this cohesive cohesin complex and ends up being stuck at that site. By simulations, we can understand what kind of structures this gives rise to. If condensin can bypass cohesin, you end up with a scenario that looks quite like what we see in experiment. We have two axes or scaffolds of condensin, and cohesin is located in between. If you take cross-sections to this simulation, you see the DAPI in blue, condensins are in the middle of each sister, cohesive cohesin is in the middle. So the complexes don't co-localize, and it's also seen in this diagram. When condensin stalls on cohesin, you end up with a very different structure actually. Now the two cores of each sister chromatid, the bases of the condensin loops, are actually stuck to each other, and cohesin and condensin co-localize. Actually, this is not at all what we see in experiment, although I must say this looks remarkably similar to a prophase, meiotic prophase chromosome. So this leads us to our second rule of engagement. Our first rule of engagement was when condensin encounters an extrusive cohesin, it will remove it. But when condensin encounters a cohesive cohesin, condensin will step over it, keeps extruding, and now cohesive cohesin ends up in the tip of the loops. Okay. Finally, we looked at the roles of individual condensin 1 and 2 complexes. And again, this is spectacular work from Komiko and Johan, making a whole series of cell lines, mostly in the background of no cohesin. So we remove cohesin in most of these scenarios. And then either look at cells where chromosomes are made by condensin 1 plus 2, or with condensin 2 only, or with condensin only, or no extruders. And we did a lot of imaging in high C on these, and I won't have time to go in any of these insights. We learned a lot about the roles of these individual cohesins and condensins. I just want to highlight one thing that I just can't get over. So happy that Bill highlighted this same contact map yesterday. But look at this amazing contact map, where we have a mitotic chromosome made just by condensin 2 in the absence of cohesin. There's one diagonal, second diagonal, third diagonal, and for some of the longer chromosomes we can see a fourth diagonal, which when I first saw this data set, I really couldn't believe it was real experimental data. It looked just, well, it's art. Anyway, so beautiful, interesting structures emerge, and it's really work from Anton, Goloborodko, who worked with Leonid for many years, now is leading his own group in Vienna. He's really a spectacular polymer modeler, physicist, and collaborator. And he has built models to try to explain any of these structures I just described and the role of loop formation in that process. I won't have time to go into it, except to say that typically, this is already an old slide, the one that Bill showed yesterday was much more impressive. The models that Anton was able to make reproduce high-C maps in remarkable ways over many length scales from kbs to hundreds of megabases. And he can reproduce these kinds of complicated structures with multiple diagonals from these models. The reason why I introduce all these things about these mutants, because I think it tells us a little bit more about how condensins interact with each other to make a mitotic chromosome. So here's a chromosome made by just, I think, condensin 2. What we learned from these models, all kinds of interesting features that I won't have time to talk about in detail. For instance, helicity is not as irregular as we had suggested a few years ago, which is a very important update to our model. We also find there is a need for small spacing between the loops, between the condensin loops. It was also seen in unrelated, independent work from the Schubert lab last year in barley. And that we also found that that cohesin between the sister chromatids put an important constraint on this helical path. And again, these are interesting details for future study that I won't have time to talk about in great detail. The most important feature of these models that I want to highlight is that in all the models that reproduce the high-C data and the imaging data from Comigo, the best is when the loops are always consecutive. Now, condensin loop being consecutive means that condensins do not bypass each other. So consecutive means that one loop ends where the next one begins. And as I said, we find, and also Schubert found, that there's even a small spacing between the loops. So that suggests that the condensins stop when they run into each other, or get very close to each other, and do not pass each other or unload. So that brings us to our final set of 3 rules of engagement. The first rule of engagement was when condensin encounters extrusive cohesin, it will kick it off and then keeps extruding. When condensin encounters a cohesive cohesin, it will step over it and keep extruding, and now cohesive cohesin is inside the loop. And the third rule is when condensins run into other condensins, they stop and hold. I'm really interested and very excited to see that you just need these 3 rules, plus an extruding machine, to explain how cells convert an interphase conformation into a mitotic chromosome. Okay? It explains how you end up in prometaphase with all extrusive cohesin removed. There's probably also an condensin-independent mechanism through WOPL that removes this. It explains how you get nested loops, how you get consecutive loops, and how cohesive cohesin ends up at the surface between the sister chromatids and doesn't co-localize at the base of the condensin loops. I find this very appealing, as Leonid always points out. How can machines that act at the nucleosome level, it just brings in the next nucleosomes as it is extruding, how can it make a micron scale reproducible structure? And I think some of these ideas we're presenting here can explain it. If you have a loop extruder that just acts locally, it has no idea where it is in the genome. It's just making loops, very, very busy, but it runs into other complexes, and these interactions need to be resolved. These 3 rules of engagement help explain how then this higher order structure to micron scale naturally emerges. I think it's a very appealing idea. Okay, so to summarize, I think I discussed various mechanisms of chromosome folding, and the number of mechanisms that we know of currently is not that great. There are only a few. But it's the interactions between them and the regulation that can explain how you can make different chromosome shapes during the cell cycle, how you can make different chromosome shapes across the tree of life. And it's really the differential control and interplay of these mechanisms that determines the outcome of folding. I just gave you one example of the transition from interphase to metaphase and focusing just on the cohesin condensant switch and how that is implemented. But obviously there are other things happening. Compartmentalization is turned on and off, entanglement is somehow controlled. So there's many other interplays that need to be explored, but this illustrates the power of such a system to study these things. Also, I just described 3 rules of engagement. I'm pretty sure there are more rules of engagement, not just between SMC complexes, but between SMC complexes and other factors. I want to highlight a beautiful paper from Romain Kozul's lab, where they showed that extruding cohesin cannot bypass cohesive cohesin. So there's another rule that they didn't call it a rule like that, but I think that's another example where the interplay between SMC complexes determines the outcome of the folded chromosome. And with that, I'd like to thank my beautiful collaborators. We saw some of these slides, my funding agencies, and all of you for your attention. Beautiful.
Thank you. You have a fantastic talk and a very good example for the other speakers, being amazingly on time. Let's have some questions.
Very cool stuff, Job. I was curious what your perspective was on what might be the most fruitful way to directly test these sorts of interaction or encounter mechanisms, whether it's going to be in vitro, in vivo, by modifying these complexes to have new behaviors, or yeah, what's next?
Yeah, I think I was very inspired, and Bill and I were talking about it yesterday in the afternoon. We heard a talk on the single molecule experiments with chromatin templates. I think would be a way to go if you can extend the length of these templates so you can see loop formation on chromatin templates with multiple complexes. I think is one way to go. Ultimately, I do think you want to see this happening in vivo. So being able to actually, for the cohesive and extruding cohesins, we have a very little ability to label one or the other form of cohesin. So having better molecular control over these complexes in living cells would be really important. Ultimately, we would love to see all these processes, of course, inside living cells happening at atomic resolution. So there are, of course, increasingly methods to do that, but I think that is what we need because, but I like to think that some of the analysis we do here leads to hypotheses that can be tested more directly and specifically. And some of these things will work differently than we think now, obviously. But I think designing experiments for a particular pairwise interaction would be really good to know. Like for instance, in vitro, condensin has been shown to be able to bypass other condensins. In mitotic chromosomes in vertebrates, it seems like in vivo that may not happen. So we're missing something in vitro, it seems. So, I mean, reconstituting the system so that it reproduces what we see in vivo would be very powerful. But I think we have a lot of talent in this group. So I think there are really important questions out there, and some of them you highlight. And I think we're going to learn more about that. But it's not going to be by Hi-C or by image. I think we need other ways to do this.
Hi, beautiful talk, thank you. I wanted to ask, if you deplete WAPL, in that case, a lot of cohesin stays on chromatin in prometaphase. So do you think that all this cohesin is cohesive cohesin, or it might be also loop extrusive cohesin? In that case, it would not be unloaded by cohesin.
It's a very good question, and we are looking into interplay with WAPL. WAPL is the one factor we all know that does unload cohesin. So is this condensin-mediated loss of extrusive cohesin dependent on WAPL? We have some evidence to suggest it is, but not all of it. If you look at where the cohesin is in a WAPL-depleted prometaphase chromosome, it is at a location where we expect cohesive cohesin. So I think a lot of the cohesin that's still there is cohesive. It's in between the sisters, but I think there's probably still some extrusive cohesin as well. So I think I have no definitive answer, but I think it's a very important question you ask, like how is this removal in the same pathway as WAPL? And I like to think about WAPL resolving complexes that are stuck and then unloaded. That could be the case. although it could also be that it just physically removes it, and that both pathways contribute to loss. We are actively studying it right now. It's a really, really important question. Thank you.
Joop, back here. That was really very beautiful. Congratulations to Johan and Kumiko and Samir on some spectacular work. So I had a lot of questions, but I'm only going to ask you two. One is functional. Only one, functional. Okay. So given that you see this massive loss in loop organization, you would predict that there'd be some pretty severe consequences for cells. Are the cells just merrily rolling along despite all these mitotic entanglements when you lose any of these four complexes? Or do you see a persistent memory of the lost state as you go through the next few cell cycles? Thanks.
Yeah, that's a very good question. So depending on which particular perturbation was done, the cells cannot exit mitosis. So for instance, many of the studies we've done here is in a cohesin depletion background, is to isolate the effects from condensins from cohesins. In that scenario, the cells cannot go through anaphase. But there are other cases when they actually do, and we're quite curious to know what happens, for instance, with entanglement resolution when you go out of mitosis.
Yeah, thanks. It was great. I'm intrigued by the rings that you see specifically at the CTCF mount site. I wonder, I mean, I'm sorry if I missed it, do you think that it is specific to CTCF mount site, or is it specific to paused loops? I mean, and do you think that tension could be at play? I mean, and would this be related to cohesin? I mean,
yeah, it's a really good question, and we did. Sunny is looking into this a little bit, and what we have in that particular paper is that it's kind of hidden, but the reviewer brought it up. It was a very good suggestion. In high C, you can see loops between CTCF sites. Those are two CTCF sites brought together by cohesin. Those interactions you lose when you cleave open cohesin. You also see flares that are anchored on one CTCF site, and now you see this flame of interactions with a second site that's not CTCF. Those are not lost when you cleave the ring. They don't depend on ring integrity. So that's kind of curious. It needs, only when it has two CTCF sites does it seem to make that conformational change. There's one other place where we find cohesin is lost, is sensitive to ring opening, and that's when it's stalled at promoters.
So you think it's posing more than interaction with CTCF?
Yeah.
So do you think there's any chance there could be handcuffs between, a handcuff model between the two CTCF sites?
If cohesin extrudes as a dimer, it might well be. I don't know. I try to stay away from the handcuffs.
Here. So how important is transcription and the fact that RNA polymerase is supercoiling and doing all that type of thing?
There is definitely an interplay, maybe a rule of engagement between RNA pol 2 and cohesin. Ed has done some nice work in that area with Jan-Michael Peters' group. Running polymerases seem to be incompatible with extrusion, is that right? Ed, you see a severe depletion of cohesin in the body of active genes. And so there is an interplay, and how supercoiling ahead and behind transcription plays a role, I really have no idea. Recent work from Jan-Michael Peters suggests there is a supercoiling aspect to loop extrusion. And Kees Dekker's lab has shown that supercoils can recruit condensins. So there's clearly some interplay between all these different mechanisms. I don't know how it works yet, but there is definitely such interest. And the fact that cohesin, I still can't go over this, that cohesin is basically regulated by all these transcriptional elements is to me intriguing. It either means these elements are really there to control cohesin, or there is just a direct link between transcriptional control and cohesin loop extrusion.
Yeah. Hi. So in vitro, cohesin is bidirectional loop extruder, condensin is unidirectional loop extruder. So I wonder, this doesn't seem to be taken into account in the modeling, or did you take it into account, and how this affect the structure in the end?
Yeah, so that is a very important thing. I think we often assume it's bidirectional, either because it acts as a dimer. In some of the models that Samir did for looking at whether it steps over cohesive cohesin or pushes off extruding cohesin. So we're a little bit agnostic as to how that actually is achieved. Either it's a bidirectional motor or it is a dimer of a pair that extrudes in different directions. For the models that Anton makes of mitotic chromosomes, he actually does not actively include the extrusion process itself. He simulates a set of loops that have been made. And then of certain sizes and with or without helical path and so on. So those are not active models that produce an extruded conformation. But I think this is another really great example where I think the collaboration between structural biologists, biochemists, and modelers is really critical now, because the modeling will change when we learn more about the biochemistry of the complex. So I think there's great opportunities here for refining these models and go beyond the static models that we have for pro-metaphase and actually see how they get formed in real time.
Do you think in either of the two categories of cohesin, whether STAG1 or STAG2 and their interactions are important, or they make a difference for this type of condensate unloading?
That's a really good question. We know that STAG1 and STAG2 cohesins have very different properties. We looked a little bit into this to see which of both complexes can end up at the C2CF sites. We really didn't see strong evidence for one way or the other, or one complex over the other. So yeah, we're a little bit in the dark about that, but my suspicion is exactly what you suggest, and that is I think the subunit composition is going to play critical roles in how these complexes work. We also looked at acetylation of cohesin, but it also doesn't really distinguish between these different conformations in our hands.
Thank you for a very nice talk. I was wondering, you mentioned the two sisters at the constraining the helical path. I was wondering if you could comment a little bit more on that. Like, is the presence of the sisters necessary for this? Can that be tested by reconstituted systems of just one chromosome? Do you get the same structure or not?
Excellent question. So the evidence we have, and that I didn't have time to include in today's presentation, is that if you have cohesive cohesin present, not extruding cohesin, it's specifically for cohesive cohesin. If you have cohesive cohesin present, the helical path is not as strong as when you don't have cohesive cohesin. As if the connections between the sister, and intuitively this is maybe obvious, but it's nice to see this in experiment, is that when you have two connected polymers and you want to spiral them independently, that's going to be a little bit difficult when they're connected to each other. So what we see two things happening if you remove cohesive cohesin. One is the signature of a helix, which is the second diagonal, gets much more pronounced without cohesive cohesin. And second, the position of the second diagonal changes so that the helical turn gets larger and the chromosomes get shorter. So that's what we know from a phenomenological point of view, that cohesive cohesin clearly affects the spiraling of each individual sister. It's not required for it. In fact, it seems to inhibit it. How it works molecularly, we don't know yet.
Thank you.
So here's the advantage of having a speaker who's on time, and also having organizers who picked a great audience. These are fantastic, it's a great discussion.
Great talk, thank you. Like, I have a curiosity, because you showed some metaphase of human cell line, and then you showed the chicken, and that is super interesting with this helical structure. And I think most of the work at the molecular level you did on chicken, but they were quite different, the two. Can you make a comment about that? And how do you think that we can extend to all vertebrates?
Yeah, it's a really excellent question, and I'm not sure. Actually, I had a slide that I removed. So the idea is actually very common to what I introduced, is that you can run the same mechanism in different ways to get chromosomes that can look different. This happens between interphase and mitosis, but also if you compare mitoses from different organisms, or when you compare mitotic chromosomes at different developmental stages in the same organism. So for instance, if you look at Xenopus, early cleavage stages, you reconstitute chromosomes in an egg extract. You get these very long, thin mitotic chromosomes. The architecture is the same. It's a loop array made by condensins. But if you take chromatin from a differentiated cell from Xenopus, put it in the egg extract, the same machine is going to now make this mitotic chromosome, but now they're very short. And it turns out you just load different quantities of condensins on the chromatin, and it's the chromatin itself that determines how much condensin gets loaded. Chromatin from differentiated cells recruits fewer condensins. Now the loops are bigger. chromosome shorter. So here's just a slide I took from a review a student in my lab wrote. If by just controlling the condensins, the loading or its interplay with topoisomerase KIF4, Bill mentioned it yesterday, you control the loading and maybe even processivity of these machines, you can make chromosomes short and fat or long and thin. And this explains some differences between the chicken and the human, but also we've noticed between, again, as I said, different developmental stages, and we know some of the proteins that actually control this, it's actually histone H1. We talked about it yesterday. So I think there's, again, this is another example where not just DNA elements control loading, it's the chromatin itself. It's regulated during differentiation. And it seems to correlate with the size of the cell. So for instance, if you compare mouse and human, mouse are acrocentric chromosomes, have very long arms. We found that in order for, in the mitotic chromosome, the mouse, the loops are much bigger to make the longest arm short enough for the size of the cell, compared, for instance, to human or chicken. So I think there's a very interesting biological meaning to the control of these complexes, to make chromosomes the right dimensions. But the intrinsic mechanism of how they're formed are always the same. Thank you.
Since lest we get hung up on differences between chickens and humans, Christine Farr showed that human chromosomes in a chicken cell are much more stable than human chromosomes in a rodent cell. So, well, actually, I think the synteny is closer between chickens and humans than it is between chickens and between mice and humans. So, chicken diverged earlier, but I guess got scrambled less. I don't know. Anyway, this is not my area. Let's thank you. Fantastic.
Thank you.
And now we have Gordana Wutz from the IMP in Vienna.
Okay, I want to thank the organizers for inviting me here and to give me a possibility to talk about my work. I am a part of Jan Michael Peter's lab, and in the last 10 years I was actually interested in how cohesin regulates chromatin organization. So I will quickly go through the introduction because it has been said already a lot, and we are going to concentrate on this part of chromatin organization where cohesin is organizing chromatin into loops. And it does so, we believe, by a loop extrusion mechanism where cohesin is a motor protein, extrudes loops, and it stops when it encounters two convergently positioned CTCF sites. Now, in these high C metrics, you can see that, as Job already mentioned, that cohesin does loop extrusion, and these loops are organized in a specific manner. They become part of topologically associated domains, or these red boxes here. But there is another feature that I really want to concentrate on, and and these are these red dots that I call corner peaks and not loops. And they can be called by different softwares genome-wide, and that's why we see these black boxes here. And now the boundaries between the TADs and these corner peaks are a result of cohesin stopping at CTCF sites, and this is because in many cases, since high C is a population study, in many cells cohesin is gonna extrude the same size of the loop at the same position, so this will result in these corner peaks. Now, if we deplete cohesin, then these structures are lost. And if we deplete CTCF, then cohesin stays on chromatin, extrudes loops, but it won't be able to stop at CTCF sites. And that's why these loops are not organized anymore in the way as in the control. And we are losing these corner peaks, as shown here in this graph, where this is the total number of corner peaks genome-wide, where upon CTCF depletion and, of course, cohesin depletion, the number of corner peaks is significantly reduced. Now, if you look at the localization of cohesin by using chromatin immunoprecipitation followed by sequence-seq or chip-seq, then we can see that peaks of cohesin are positioned at the same time at the same position as CTCF peaks. And these peaks are gone if cohesin is depleted. And now if CTCF is depleted, we believe cohesin is distributed along the genome, and it does not accumulate any more of CTCF sites because there is no CTCF to stop it there. Now, cohesin is a multi-subunit complex composed of SCC1, SMC3, SMC1, and STAG. But there are many regulatory subunits in G1 cell cycle stage that are controlling cohesin. One very important one is NAPBL, which is essential for keeping cohesin on chromatin, and it's also essential for cohesin to perform loop extrusion. Now, cohesin can be released from chromatin, and there are two factors that are called PDS5 and WAPL. And I would like to mention another protein and one cohesin modification that is going to be important for this talk, and this is acetylation of SMC3 subunit that is performed by two acetylases, ESCO1 and 2. Now, in mammalian cells, there are often two isoforms for the same protein, like in the case of STAG, PDS5, and ESCO, and we are going to deplete both isoforms because they are partially redundant to achieve the complete phenotype. I'm going to concentrate in this talk on PDS5 and show you the interplay between PDS5, NAPBL, WAPL, and acetylation. So all these proteins have to be tightly regulated because any perturbation of these regulatory proteins, it's also going to affect the genome organization, which is, as Job said, important for transcription, VDJ recombination, and so on. So we have already studied PDS5 function by using RNA interference. However, we could not deplete proteins completely, so we went to use auxin-inducible Dekker system in HeLa cells, and we have generated these three cell lines, WAPL, PDS5AB, WAPL, PDS5AB, AID, and we can now synchronize cells in G1 cell cycle stage where we deplete the proteins and we still collect them here, so we do not interfere with cohesive function of a cohesin, which is going from S phase to the anaphase onset. From these high C matrices, you can see, and it has been published already some time ago, that if WAPL and PDS5 are depleted, or PDS5 are depleted, so they are releasing factors, now if they are depleted, cohesin stays on chromatin longer, and it cannot be removed, it accumulates there, and for that reason we believe it extrudes longer loops, as shown here. Now, there is a main difference between these two proteins in terms of the phenotypes, and this is that upon PDS5AB depletion, we are losing corner peaks, and this is not rescued by a WAPL co-depletion. So in the case of WAPL depletion, we are actually gaining more corner peaks compared to the control, but in the case of PDS5 depletion, we are having less than in the control. And also, just to mention that the insulation shown here in this insulation plot is going down. It's going down very much in triple depletion. Now, the main question of this talk is going to be, why are boundaries and corner peaks reduced upon PDS5AB depletion? And to answer this first question we asked, are the CTCF levels still the same? Because if CTCF is not there, then that could be the reason why cohesin does not stop at CTCF sites. And we looked at the ChIP-seq of CTCF. Here you see one genomic region, and this is a quantitative ChIP-seq. You can appreciate that upon PDS5AB depletion, there is not big difference to the control. However, if we deplete CTCF, we completely lose these peaks. Now, here is the genome-wide analysis where we do a pile-up of CTCF at CTCF sites, and we can see there is not a big difference. However, if we look at now at cohesin, then we can observe that PDS5AB depletion results in smaller peaks at CTCF sites. Not as small as upon CTCF depletion, but still significantly smaller. And this we can see in this pileup of cohesin at CTCF sites. And now in cohesin, now in PDS5AB depleted cells, we have actually more cohesin. But at CTCF sites, we see reduction. So where is this other cohesin positioned? It's very often positioned at promoters and convergently at the sites of convergent transcription, as shown here. So it accumulates there. And if we look at the genome-wide analysis of cohesin at non-CTCF sites, then we can see increase of cohesin upon PDS5AB depletion compared to the control. So the CTCF levels do not change upon PDS5AB depletion, but cohesin does. So, so from this we can conclude that there must be something about cohesin property that is enabling cohesin to step over these CTCF sites. And first we ask, is this the residence time? Could it be then upon PDS5AB depletion, cohesin gets stabilized so much that it can cross CTCF? And for that, we have performed experiment which is called a fluorescence recovery after photobleaching, where we indeed bleach half of the cell, and then we look the difference between the unbleached and bleached region over time. And then we can see in the control cells that this recovery happens completely upon 40 minutes. However, upon PDS5AB depletion and WAPL depletion, we can see stabilization of cohesin. However, interestingly, now the stabilization of cohesin upon PDS5AB depletion is much less than in the case of WAPL depletion. So this cannot explain why cohesin crosses CTCF. So the next question we ask is acetylation actually is required for the formation of corner peaks? And here I just want to mention that PDS5 A and B localize by ChIP-seq very often at the loop anchors, and this is also the case for acetyl SMC3. And they do so much more often than it's the case for CTCF and cohesin, meaning that many of CTCF and cohesin peaks are localizing somewhere else and not at the loop anchors. So there is some function of PDS5 and SMC3 at the loop anchors. Now, PDS5 is not only important for the release of cohesin, but it's also essential for cohesin to get acetylated in G1 cell cycle stage. So without PDS5, ESCO cannot acetylate cohesin on chromatin. So to look at the function of acetylation, we are going to deplete ESCO1 so that we don't interfere actually with the releasing function of PDS5. And we did so in the collaboration with Dana Branzai's lab, where the ESCO1/2 AID cell line was generated. And what we do, we again synchronize cells at auxin and harvest them in between S and G1 phase, and then then perform high C. And what you can see here is that upon ESCO1/2 depletion, interactions become longer. So that means that acetylation is indeed inhibiting loop extrusion, similarly like WAPL or PDS5 too. However, the acetylation is not essential for corner peaks because we can see even slightly more corner peaks upon depletion of ESCOs compared to the control. And moreover, if you now look at the localization of cohesin by ChIP-seq, we can see the peaks are very similar to the control peaks. So we concluded that SMC3 acetylation is not essential for formation of corner peaks. So what could be another property of cohesin that enables cohesin to bypass CTCF? And we wanted to ask if there is a composition of cohesin that might contain more NAPBL, because in that case, cohesin would be more processive, because I told you that NAPBL is important for loop extrusion of cohesin. And could it be that PDS5 competes with WAPL? And this question has been proposed already in the field, because PDS5 and NAPBL have very similar structures, as shown here, but they are crystal structures. So it's predicted that these two proteins could bind to the same site of cohesin. However, they have very different functions. NAPBL activates cohesin's ATPase activity, and together with cohesin can perform loop extrusion in vitro and in vivo. However, PDS5 does not have this ability to activate cohesin's ATPase activity. And then if we incubate cohesin together with PDS5 in vitro assay of loop extrusion, we cannot see cohesin performing loop extrusion. So what we wanted to see now, what is the composition of cohesin in different scenarios? And for that, we have used SMC3 antibody to IP the whole cohesin complex. And then we have submitted those, samples to, for the quantitative mass spec. Here you see the isolation of immunoprecipitated complexes. And then the resulting peptides, we have normalized to the levels of SMC1, 3, and SCC1 core cohesin subunits. So that's why you see the levels of SMC3 peptides the same. However, of course, upon PDS5 depletion, we can see that the levels of PDS5B in this case go significantly down, and in the case of WAPL depletion, WAPL signal goes down. And the interesting thing is that now we could see more NAPBL bound to cohesin complexes resulting from PDS5AB depletion compared to WAPL depletion or the control, 2 to 3 times more. So we believe that we now have more cohesin complexes occupied by NAPBL. And moreover, and this actually goes along with the idea that PDS5 and NAPBL compete for the same cohesin part to bind. Moreover, we performed also, again, IFRAP analysis, in this case for NAPBL, in different depletions, and we could observe that NAPBL gets more stabilized upon PDS5AB depletion than that's the case for WAPL or control. And we could even calculate the residence time from these curves. So now we are ending with the cohesin that has more NAPBL, and this NAPBL is more stabilized. So could it be that this cohesin is now more processive, and this will enable it to pass CTCF sites? And to really answer these questions, what we did now is we have these PDS5AB cells where we can deplete PDS5AB, but we can also partially deplete NAPBL. And we just went to look if we can see now more corner peaks in this situation. I have to say that this experiment was done very carefully in the way that we started to work with the same number of cells. We have the same number of reads here. And what you can see that I hope that the corner peaks become now stronger if we partially deplete NAPBL. And moreover, all the structures that are very fuzzy upon PDS5AB depletion, they become now stronger in this situation. And the number of peaks goes up. Moreover, if we now perform ChIP-seq in these different situations, so you see in the control cells, then upon PDS5AB depletion, these peaks go down. However, if we now partially deplete NAPBL, at least some regions, it looks like that there is more accumulation of cohesin at these sites. We also wanted to ask how PDS5 and NAPBL work by using, in single molecule studies, by using in vitro loop extrusion assay that has been established in our lab for cohesin by Ian Davidson. And what you can see here is if we have a perpendicular buffer flow, then we can see this loop is extended after we add cohesin, ATP, and NAPBL, of course, to DNA. We can also do this experiment without buffer flow, without perpendicular flow. So then we can see that this loop is here, but it's like squashed. And from the intensity of this loop over time, we can actually measure how much DNA is incorporated into this area here. So for example, here we will have more loop extrusion going on, and at this point it's going down. Now we can label NAPBL, and we can almost always see that NAPBL signal correlates with the loop extrusion. So loop extrusion is still here very strong, and NAPBL is always there, but it comes off when the loop extrusion starts to slow down. And interestingly, not always, but in many cases we can see that PDS5, if we label it, comes exactly at the time when NAPBL leaves DNA. So we do believe, or we would like to believe, that actually this experiment shows that PDS5 and NAPBL exchange, and that during NAPBL binding to cohesin there is a loop extrusion, and when PDS5 comes, then this loop extrusion slows down. So what I told you today is that we believe, like many of us here, that NAPBL together with cohesin extrudes loops, and when it encounters convergently positioned CTCFs, then PDS5 exchanges for NAPBL, and also acetylation happens there, and this enables then cohesin, or this leads to cohesin stop at CTCF sites. So this has been already published by two other groups, Roland and Beckwith. And what I told you today, that actually PDS5 has also a function of regulating NAPBL levels on cohesin, and if PDS5 is not there, then we get more NAPBL on cohesin. This NAPBL is more stabilized, and this will lead to cohesin bypassing CTCF sites. So I would like to acknowledge the whole lab, especially my boss Jan, who actually allows me to do whatever I want. Here are the people in the bold who contributed to this work. And thank you for your attention.
Thank you very much. That was great. The microphone is slow coming from the back, so I'm not going to throw it.
Okay, thank you. That was a beautiful talk, and I think the results are amazing, but it's interesting to think more in a dynamical way, that these loops are not actually formed 100% of the time, they're actually opening and floating, and that's sort of, I have a couple questions. One is when you look, for example, a lot of the peaks, cohesin peaks into CTCF formation, they have different intensities. That means that you may have different numbers. Should we believe that we can calculate how many cohesins there? And also, when you look at your WAPL data, what's very nice is that yes, you have these longer loops, but they're actually loops of loops, right? If you look at their position, they're bringing two
nested
loops, and
loops,
they're bringing two loops together. So is this something where all these loops are formed at the same time, or some of them are formed part of the time, and this cohesin is sort of moving back and forth? That's what
So in the case of WAPL depletion, it's interesting that cohesin becomes very much stabilized. So it can stay, some of cohesin stay like for 6 hours or longer.
Right, but they're moving back and forth. There's a...
Well, I am not so sure. I think in some situations, in some cells, you get one loop, and then in the so because upon WAPL depletion, cohesin stabilizes on chromatin, and it can also stop bypass CTCF site,
exactly.
and then it will. But for some reason, it will stop at another one. So that's why you get longer loops.
So you think they always move in one direction? And because these loops are somewhat open and closed, I think it's beautiful because if you really look all the additional loops, they are longer loops, but they're not like anywhere. They're really bringing two other loops together, right? The position is very...
Yes, and actually all the corner peaks have kind of a similar strength. So that would...
And then that's nice because you're discussing processivity, right? So for example, if I look kinesin, it's very easy to understand processivity, but like I think we heard his talk yesterday, these motors are very weak. So the question is, you need multiple motors to help you to get those loops formed, and I think results are very nice. I have to think about it. I want to know what you...
Yeah, well, I don't know about this question, how many cohesins are present there. From our in vitro studies, we believe it's one cohesin molecule that extrudes loops, but of course, we don't know what's happening in cells. Yes.
Okay, thank you. Yeah, Gordana,
yes.
Very nice talk. And I have a question about this single molecule experiment. Does presence of PDS5 extend the periods when NIPBL is not bound, right? The periods when the loop is going down, or does it make the stretches when the loop is growing shorter? Can it outcompete NIPBL? You see what I'm saying? So which parts change when you add PDS5 to the solution?
Well, we can sometimes see PDS5 bound to cohesin and traveling together with cohesin and NAPBL. So, but of course we don't have always single molecules, so it's very difficult to get really great conditions. It also depends on the cover slips, how clean they are. So it's very, very difficult to, very often we see a strong signal, and that would indicate there are many cohesin complexes stuck there. But in some cases we do see single molecules, and in these cases we tend to believe that actually in those cases PDS5 comes and displaces NAPBL and loop extrusion goes down.
Right. And what's also interesting from this trajectory is that the loops, that when PDS5 is bound, the loops are not stable. They're getting smaller.
Exactly, yes.
But cohesin does not dissociate. The trajectory continues because then NIPBL comes to the rescue.
Yes.
So it's not that when PDS5 is bound, it lets the loop go, the loop stays. It might be the effect of the flow, though. So I wonder whether it's the same.
Of course. So I think that if we believe that cohesin does stop and hold the loop, that is much more complicated. We would need to have a CTCF in the system, and we might also need to have acetylation in the system. And this is what we want to look at in the future. Yeah. So here we just wanted to see, does PDS5 displace, or can we see, can correlate this with PDS5 NAPBL exchange?
That was really beautiful, Gordana. Congratulations.
Thank you.
So I really love that FRAP experiment where you looked at the altered residence time. It's actually quite striking. And I had a question that was kind of similar to Jose's, but I thought of an experiment. And I wondered, given that you're in Jan Peter's group, can you tag random loci along with the FRAP experiment where you're tagging the proteins, and then look at this dynamic formation and loss of these little loops in vivo, which would very nicely couple to your in vitro, gorgeous work, because that would give you a real insight into whether that acetylation, the time scale at which this is occurring, right?
Yeah, so we have not started doing these experiments, but that would be very interesting, I suppose.
Wonderful
Hi.
talk.
Hi.
Could we see the cartoon on the previous slide for a second? So I was curious, do you have any insight into whether this acetylation is happening before or after the PDS5 binding? And if regardless, is there an experiment to distinguish sort of the order of operations at the CTCF on that left-hand diagram?
Here on the left, yeah. Or? Well, I don't know how the events occur. What I know is that if we deplete PDS5, loops become longer. If we lose acetylation, loops also become longer, but not as much as in the case of PDS5AB depletion, because PDS5AB is also important for release. So we get smaller loops compared to the PDS5. And what is also interesting is that acetylation and PDS5, so we might think that this stabilizes cohesin. And this is also interesting that it's dependent on CTCF. If we don't have CTCF, we won't see acetylated cohesin. So all these three things come somehow magically together. We don't know how does that work. So that's the only thing I can tell you.
Okay. Oh, hi. So,
hi.
So I just wondering, is all the cohesins either bind to the PDS5 or NPL or WAPO in the cell, or actually the sort of regulatory things is?
Yeah, okay, so that's an interesting and complicated question. Okay, so I can tell you what I believe. So I do believe that that NAPBL and PDS5 compete with each other, so that there will be a cohesin bound to NAPBL and cohesin bound to PDS5. Now with PDS5 WAPL, it's a more difficult question. So it has been proposed in yeast that PDS5 and WAPL are binding partners. So then basically, if you deplete one, the other will also come off, and that's going to be a release. Basically, both are important in the same pathway to release cohesin. But what I personally believe in human cells, it's a bit different, because we have evidence, I have not talked about it here, that actually PDS5 might release cohesin in a different way than WAPL, because WAPL opens exit gate, but we think that also PDS5 can exchange for NAPBL at some complexes, and that, for example, are not acetylated, and they can get released then from chromatin. So that's my so we do believe that PDS5 has also partially independent WAPL release.
Okay, thank you very much, Gordana.
Thank you.
That's another stimulating talk. And now we have a brief announcement from Ed.
Yeah, just two quick things before the coffee break. So first, if you're doing the social outings for the Freedom Trail, please meet downstairs at 1:30 for the museum or the...
Okay, so let's start the next part of the session with Héloise Muller, who's going to tell us about holocentric chromosomes.
All right, so hi everybody. So today I'm going to tell you about the genome architecture in the silkworm, mainly Bombyx mori. And maybe the first question that we can ask is why do we want to study this species? And here is some of the reasons. So first, since approximately like 20 years or so, people have been sequencing a lot of animal genomes, but we can see from this recent, review that there is a huge overrepresentation of genomic studies in chordates. While actually the biodiversity mainly lies in arthropods and more specifically in insects. So that's why we got interested in this group of species. Among insects, we actually started our study with Lepidoptera, which are butterfly and moth. And obviously one of the reasons is because some of them are really beautiful, but not only. It's also because of this H that you can see on the branch going to Lepidoptera, and also in 3 other locations on this tree, meaning that among insects, there has been recurrent and independent transitions to species that become holocentric. So what does holocentric mean? Actually, most known and studied organisms are monocentric, meaning that they have only one centromeric region per chromosome, which is at the construction point between sister chromatids on metaphasic chromosomes. And here, actually assembles a protein complex called the kinetochore, which will link the centromeric chromatin to the spindle during cell division. And Lepidoptera actually have an alternative centromeric organization where the kinetochore complexes actually assemble all along the length of metaphasic chromosomes, which have no constrictions. So with this, you will have multiple attachments to the spindle, and the species with this configuration are called holocentric. Then, with the studies that have been done mostly in monocentric species, we also know that centromeres are actually really strong anchor points in the 3D organization of chromosome inside the nucleus. So with this, we actually wondered if this alternative organization was actually impacting the 3D organization of chromatin inside the nucleus. So we initiated the study actually working on the silkworm, Bombyx moe. And the reason for that is that this is actually the reference organism in the insect Lepidoptera. And also because at the time when I initiated this study, it was the only Lepidoptera that had a great fully assembled genome with full-size chromosomes. So if we shortly review this genome, it's approximately 450 megabase pairs, and it's organized in 27 autosomes and 2 sex chromosomes. The chromosome Z, which is actually the chromosome 1, and the W is actually unassembled here because it is full of repeats. It has approximately 4,000 protein coding genes, and approximately half of the genome corresponds to transposable elements. This genome is actually really poor in other type of repeats like satellites, except that for the telomeric repeats. And finally, we see that the mean GC content is approximately 38%, but this corresponds to also some heterogeneity between chromosomes, and even more specifically, within each chromosome where the large subtermeric regions tend to be higher in GC than the chromosome centers. So to actually go more deeply in our subject, what do we find doing high C on this organism at different levels of organization that has been described, mainly using mammalian genomes then? So first, at the genome scale, the Bombyx chromosome have actually really strong chromosome territories with really sharp demarcation of contact frequencies between chromosome in cis between chromosomes and compared to trans between different chromosomes. And this is actually really similar to the textbook example that is shown here, but it's really different than what you see in most organisms. And as a comparison here, I draw the map for fly chromosomes. And in this case, if I didn't plot this dashed line, it would be really hard for people to actually find the border between chromosomes. Additionally, if you look at the trans contact area here, as has been described in also many species, you can see that you have other specific type of contact which mainly correspond to centromere-centromere clustering. And this is absent from the trans contact area in Bombyx, and probably consistent with the fact that it is holocentric. Then, on the intermediate organization, at the compartmentalization level, we actually describe peculiar organization in Bombyx. So we actually do find classical active A and inactive B checkerboarding and compartmentalization. But in addition to that, we find regions that do not are not involved in any checkerboarding and actually make really frequent contact near the diagonal and very little contact further away at longer distances. And we actually describe this as a third compartment that we call S for secluded. So now if we go to the finer scale at the TAD level, so we can actually see tide-like structures in the genome of Bombyx mori with everything that you could find like triangles, corner dots, flames, etc. So with this, we could actually describe all the different levels of organization in Bombyx mori, but we got mainly intrigued by these peculiar regions that we want to study further. So for that, here I'm actually showing you showing this in the context of a full chromosome for you to get a better idea. And again, we can see some checkerboarding and some regions that actually do not take part in this checkerboarding. So then I wanted to actually describe these three types of chromatin organization in each chromosome. So what I did for that is draw the Pearson's correlated map from this and extract the first three PC vectors. On this, I then performed K-mer clustering to actually find 3 clusters per chromosome and attributed them to A, B, and S according to their epigenetic profile that I will describe later. So with this, you can see that these S regions actually corresponds mainly to the white stripes in this matrix and are often positive in PC2. So we did that analysis genome-wide, and we can actually find S compartments on all the different chromosomes. And taking them all together, they actually represent approximately 15% of the genome. If we look at the size distribution between different domains of each compartment, you can see that S and A compartment domains tend to be slightly smaller than the B domains. But in S, you can also find some really large outlier domains. Okay, so now let's take a look at how I actually characterize these compartments. For this, I use chip tracks from common epigenetic marks. And so I used H3K36 trimethyl and H3K4 trimethyl as mark for active chromatin, and H3K27 trimethyl and H3K9 trimethyl as mark for inactive chromatin. Then I also included this mark here, H4K20 monomethyl, and this mark has been implied in actually many different processes, and we originally included it because it has been shown to be associated with centromeric sequences in some papers. So now let's actually look at the genome-wide enrichment of these chromatin marks in the 3 different clusters that we have. So for the first cluster, we can see that we have a strong enrichment in active marks and depletion of inactive marks, especially H3K27. And so we call this cluster compartment A, just for their similarities to other species. And then the 2 others are actually depleted in active marks and slightly enriched in inactive marks. So we actually call compartment B the compartment that was involved in the checkerboarding, again to be similar to what people have described in other species. And the other compartment is the compartment that we call S, and it actually corresponds to this peculiar region that we described as non-checkerboarding. And it has mixed epigenetic features of A and B with depletion of active marks and some enrichment in active mark, but also an enrichment in H4K20 monomethyl. So then we wanted to actually dig into the possible biological relevance on this compartment. So for this, we start to analyze the sequence that were in this S compartment. So in general, we can see that S compartments are GC poor and also depleted in transposable elements, most similar to a compartment in this case. But their main characteristic is actually to be very gene poor and actually often correspond to gene desert. Yet, if we look at gene expression doing RNA-seq, we can see that these genes can actually be expressed, and they are expressed at similar level as the genome-wide expression. If we dig a bit deeper into these genes, LRNS, that's approximately 300 genes, and do some gene ontology, we can see that they correspond to transcription-related and DNA binding proteins. So in our mind, this could indeed link to possible transcription factors. To actually dig into the possibility of biological function in this S compartment, we also took advantage of another property of this S compartment, which is their plasticity. And we actually saw that they can change during the development. So for my initial analysis, I actually focused on one embryogenesis time point, which is the one in the middle here, which corresponds to 24-hour post-diapause. But I also did experiments earlier in early embryos and embryos at 2 hours post-diapause, and experiments later at 48-hour post-diapause, and a drastically different data set with adult height. And on the top here, I'm just showing you an example of a compartment S that actually appears between the time points and is then maintained in the adult head. And if you make these differential maps to compare between the 2 hours and 48 hours after diapause, it shows as a red stripe here. And on the bottom, I'm showing you an example of the reverse, which is a compartment that was corresponding to S domain, and then is turning to a checkerboarding as a B compartment, and also is maintained in adult heads. And this is shown here by a blue stripe. So to actually be slightly more exhaustive on this, I tried to compare the two extreme time point, which was the one that I focused on originally, and I tried to look at the becoming of compartments called at this 24-hour post-diapause in adult heads. So doing this, you can see that compartment A and B are mainly stable, while only half of compartment S are maintained, and most of the one that changed then turned to B. So we try to take advantage of this and dig into some of these compartments that actually switch. So this is an example of one of these really large S compartments that we have in the embryo. And here is a close-up of this region. And you can see again, as Emily has been showing you yesterday, that inside this S compartment, you can find many structures like insulation points, dots, etc. And some of the genes that are inside, even though there's indeed low gene coverage compared to other compartments, can be actually expressed by RNA-seq data. And some of the genes might also actually correspond to some of the borders that we observed. Now, if we look at this region in adult heads, we can see in the chromosome context that this region is now totally checkerboarding. And if you look at the close-up, we are indeed losing a lot of the structure and gene expression. This can be more generally recapitulated by some boundary pileups. And you can see here that compartment S that actually switched to B now have a boundary of lower strength. And similarly, if you look at gene expression, you can see that compartments that switch to B usually have a drop in gene expression. To show another example of this, I put here one of the other really large S domains of the genome on chromosome 6. And we think this one is of particular interest because it corresponds to the Hox cluster in the bombyx. And in embryos, we believe this OX cluster is active and has a really important role. And we can see, again, triangle, corner dots, etc. in the structure. While in adult heads, we lose most of this structure and gene expression. So from this, we actually suspect a role of this compartment in gene expression regulation in general, probably in the development or more generally in cell differentiation. But after doing all this, there was actually one question still pending, which was how are these compartments mechanistically formed? And for this, we have had the help of Leonid Meny's team, and more specifically, Emilie Navarrete, that maybe you have seen yesterday, but as a poster here, if you missed it, and you can go back to her. So I'm not going to explain fully the model, just saying that indeed she based her simulation and agreement with actual observation on three main characteristics of S compartments, which is first, they show enriched contact close to the diagonal within contiguous domains. Then, between two S domains, there is a depletion in compartmental contact with other S domains. And finally, there is a depletion of contact between S domains and all other compartments that is homogeneous, meaning like we have smooth lanes of depletion. So just to make this story really short, I'm going to tell you just the main result of all of this modeling, which is we think that the unique A, B, and S pattern that we see in Lepidoptera actually requires both mechanisms. First, homotypic attractions among monomers, which is driving compartmentalization, and a basal level of loop extrusion all along the chromosomes with a higher level of extrusion in S. And so with this model in mind, we actually went back to the data and tried to see if we could find evidence of this. So one of the things that we can do to analyze the high C data is actually use this scaling plot that you've heard of. So they correspond to the probability of contact according to the genomic separation. And this curve, actually, when it deviates from a linear line, it's because it actually deviates from an homogeneous decrease of contact. And it means that some structure at specific separation are actually making more 3D structure than expected. And to study this shoulder in the P of S, we often actually draw the derivative. And on this derivative, people have shown that this shoulder actually then look like peaks, and that this first peak has been mainly attributed to the action of loop extruder, in particular the cohesin, and that the local maxima actually corresponds to the mean size of the loop. And so this plot is actually indicating of some loop extrusion occurring in Bombyx-Maury genome with a mean loop size of 40 kb. Now, if we dig into this and draw the same plots within each compartment type, we actually can see that this loop extrusion feature is more prominent in the S compartment, with an average loop size of a similar size as the genome-wide. And we can also see some evidence of loop extrusion in B, but probably with slightly bigger loop size. And another way to look at this and to somehow explore the compactness of compartment is to look at this observed over expected pileups, again on intra-compartments, so in other words, like cis on diagonal. So with this, you can see that in A compartments, you have a decrease of contact that is actually similar to what you expect according to distance, while with B and S, you actually have enrichment of contact, and specifically in S compared to expected. So S compartment seems to be more compact than A and B. And finally, with this type of parallel plots, we can also try to explore the strength of compartmentalization by exploring, in this case, the intercompartment average. And here we can see that AA interactions are actually higher than BB, while S is actually depleted in contact with all the different types, including other S compartments, and AB is actually neutral. So all of this is actually consistent with the model, and we want to explore more of this if we can actually validate more some of the findings. And for this in particular, we would like to validate the uneven distribution of SMCs along the genome. We would like to identify maybe insulator proteins at the various type of borders and understand the functional role of S compartments. And for this, we will actually turn to a new model, which is the pentramorph called Plodia interpunctella, because now we will be able to do some genetic manipulations in it. That's the model that the Lepidoptera community is bringing forward and already showed that some manipulations are possible. So we're really counting on it. So we're already starting the rearing of this animal in the lab, and as you can see, it's pretty easy, grow in some Tupperwares, and you can have them also have a really great system of synchronized egg laying. So we started some easy manipulation of them to show that we can do some imaging of early embryos. And we also can obtain our very first map of Plodia, which is very encouraging because it shows these regions that really looks like a S and turns to be the OX cluster in this genome. So that's really encouraging for us. And with this, I mean, I'm not going to recapitulate all our findings, but I want to say this is just the beginning, and we hope that we can do a lot more with this new model, and also finally maybe explore if S compartment could be related to centromere or holocentricity, which we couldn't really tackle so far. And with that, I want to acknowledge people who did the work, and mainly the students, in particular Jose, who did his PhD on this project, and more recently Noe, who actually studied the rearing of the Plodia, and Apo, who is exploring comparative genomics to explore the possibility of this compartment in other species. And importantly for today, Emily, that did the modeling in Leonid's lab, and that, again, you can go and talk to her poster. Thank you.
Thank you, Héloise. Yo.
Wonderful talk. Thank you for that. When I looked at the chromatin traces in the beginning of your talk to explore what S compartments could look like, I had a sense there is K27 tri and K20 monomethylation, but they often don't co-occur. So I was wondering, could there be a subdivision within S of different types of S, maybe K27 versus K20?
Yeah, I mean, yeah. Let me maybe put that back on. Indeed, we are showing this like, is it coming? Oh, yeah. This overall enrichment, but they are not homogeneous across S compartments. It's indeed like sometimes if you look at this profile, it shows on some of them, not all across. I think it's a correlation, but it's not what defines these compartments probably in the end. Yeah. I mean, and we still need to know what defines them.
So, Magda?
Yes, very nice work. I just wonder if you ever looked by fish at the S compartment, how it would look like. It's so depleted, I just wonder how in 3D it would look like and where it would be localized in the nucleus.
yes, that's something we have explored. Not fully, but we have already some hint into that, which is pretty interesting. We did this work in collaboration with Leah Rosin in the NIH. And what she did is label all compartment types in different chromosomes, A, B, and S, and look at their localization relative to the chromosome territories. And what she sees is that these S are actually enriched at the periphery of chromosome territories. And this, if you heard Emily yesterday, is also consistent with the modeling that they brought, which they showed that extrusion in this compartment could be enough of a force to actually drive this relocalization. And it's also consistent with the contact pattern of this S compartment, which, although they are really depleted inside chromosome, in cis, they seem to be enriched in trans.
Oh, okay. Thank you.
Yeah, many there. Yeah.
Bill?
Yeah, please.
Hey, very beautiful talk. Sure.
I don't think we'll call names.
Oh.
Yeah, go.
Yeah, okay, sorry. Confusion. So in the slide where you showed the emergence of one of the S compartments during development, it looked like it basically emerged right next to another one, but they didn't merge together, so there should be a boundary between them. Do you have any idea what that is?
I wish. I think that's going to be my short answer. Yeah, indeed. Yeah, you would think that these two should be the same, right?
Yeah,
but
exactly.
indeed they are separate. We see like maybe two triangles. And no, for now, I mean, I would like to investigate the different type of borders. I do want to believe that maybe the borders that are at the outside of this compartment may be different from the borders that are actually building the TAD-like structures inside the compartments. And yeah, we have some clues on which protein to look at as a starting point. Of course, we're going to look at CTCF. We're going to look at other insulator proteins that are known in Drosophila, and only a few of them are actually conserved in the Bombyx genome. And maybe we can explore also more. Yeah, thanks.
Thank you. Here. Thank you for a very nice talk. I was wondering, maybe I missed it. Did you investigate if lamina is playing any role in S compartment localization?
So I did not directly do that. I wish I also do. So far, what I can see is that with the microscopy study that I showed, we do see this specific localization on the outside of chromosome territories, but we couldn't find a specific localization on the outside of the nuclear, near the nuclear periphery. So, yeah.
Bill, I have one last very quick question.
The last, I have a question too.
Oh, good. You go first. Quick question. So beautiful work from Ines and Leah and you. Congratulations. Daniel Zilberman, when he was at UCLA, published a Bombyx-Maury DNA methylation map, which strongly correlated with the end of diapause, turning down specific gene transcription program. Are you looking to maybe knock out DNMT3A, 3B, or MET1 to see if the S compartment is dependent on the loss?
I mean, all of this would be really interesting indeed. I mean, hopefully, already now we're turning to this new model, so we'll have to redo maybe this analysis in the other model, but we have, it's high possible that we'll have similar things in this one. Although, they do not undergo diapause, but maybe still the onset of setting the different compartments will have similar things. So yeah, it would be really interesting. Yeah, but so far I don't have any link to DNA methylation of this type of data. Yeah.
Thank you, and then I'll take the pleasure of the last question. So when you see these really tight knots on the diagonal, makes me think, it's the same kind of question as Magda, but asking something different, not like where they are, but have you used oligofish to try to ask, are these actually more compact areas? Have you looked at the level of
we
DNA
looked into
compaction?
that. I don't have any of the results here. It's not so clear. I mean, because you need to label really many different compartments, and we don't have this many data yet. And also like in the ideal world, label things that are on the same genomic size, so you can really compare the actual dense size on the microscope. So it's not so trivial, and we wish we investigate more into that as well. Yeah.
Thanks very much. All right, let's move on to Geoffrey Fudenberg. Thank you.
Okay, so yeah, thanks to the organizers for putting together such a wonderful event. This is unpublished work. Feedback is very valuable, especially as you will see, we draw very deeply on observations and insights from members of the audience. Maybe about half of my slides will refer to Wutz 2017. So thank you for obtaining such beautiful data on chromosomes. So in interphase mammalian cells, as has been discussed, we now believe that cohesin is one of the main genome organizers, and it organizes this dispersed sea of meters of chromatin inside a micron-sized nucleus, as seen in this EM image, and it does this by the process of loop extrusion. One thing that hasn't been introduced yet, this conference, is that we simulate loop extrusion. So we typically simulate loop extrusion by alternating beam steps on a 1D lattice and 3D polymer movements. So the 1D lattice keeps track of where extruders are along the genome, and as time progresses, extruder leg positions are updated, and extruders like load and load across the genome. The positions of these legs are then used to hold together some distal beads in 3D polymer simulations. Updates on this 1D lattice are determined by a set of parameters. Those just describe the dynamics of loop extrusion, the velocity, separation, and lifetime of these motors. From there, we generate large ensembles of 3D conformations, do things like in silico HiC, and then we often extract these contact frequency versus distance curves to compare with experimental data. The agreement of these curves in wild-type data sets and after depletion of extruders has provided some of the strongest support for genome-wide activity of loop extrusion in vivo in interface. Okay, so it's very, very useful technique so far. At the same time, in vitro measurements provide this incredible glimpse of the microscopic dynamics of active loop extruders. So this complex can processively enlarge loops at the rate of thousands of base pairs per second. This is beautifully illustrated in these sorts of doubly tethered movies that Gordana also introduced already, here showing extension and then generation of loops. And this barbell cartoon that we've been using to kind of describe all our models is, of course, an incredible simplification. And I think, in my opinion, limiting ourselves to this sort of cartoon is one of the main bottlenecks to gaining deeper predictive understanding of interphase loop extrusion. So as depicted in this more detailed cartoon, and as also previously introduced, but I'm going to remind you right now, we have this core complex with long 15 nanometer arms, and then we have these associated regulatory proteins, WAPL, PDS5, NIPBL, that I'll ask you to focus on. And they all thought to bind along this flexible linker. They're all important for the dynamics of the whole complex. NIPBL in particular is essential for ATP hydrolysis and translocation. As you just heard, PDS5 seems to inhibit this ATP hydrolysis, which has been described as competing with NIPBL. Okay? At the scale of the whole genome, what's really remarkable is that perturbing levels of even a single regulatory factor can have these dramatic, beautiful consequences. So on the left is cohesin image in an unperturbed scenario, and on the right, after depletion of a single factor, WAPL. And what we see is the emergence of these thread-like structures made out of cohesin, which the authors term vermicelli. What we now understand this is as the lifetime can be increased, and you can start to have collisions between extruders, and this can self-assemble backbones of chromatin, similar to what's happening in prophase. And at the same time, just as increasing the lifetime, the fraction of bound extruders also increases upon WAPL depletion. So just remember that if you can, because that turns out to be very powerful information that we'll use later on. Okay, so how does perturbation of this microscopic interplay and dynamics of these accessory proteins modulate the genome-wide organization? There's no direct connection right now between these two worlds. How low does WAPL need to be to create vermicelli? Do we need a certain abundance of the core complex to create vermicelli? What about predicting these incredible changes that we see in HiC maps right here? Answering these sorts of quantitative questions requires quantitative models. So we need to turn our description of this process, like PDS5 competes with NIPBL, into equations that the computer can understand if we want to make predictions. And really different formulations of the same words could lead to different equations, different predictions. So this is work with Max Tortora, a postdoc in my group. He's starting to be on the lookout for positions. He's an incredible colleague. Be on the lookout for him as well. So if we could take the microscopic abundance of dynamics of cohesin, use it as inputs to loop extrusion simulation, I'd argue you could make such predictions. Really, so the question reduces to building a map, and the map is between the set of subunit abundances and the parameters of the loop extrusion process. But it's not obvious what should the velocity be, the lifetime be, the separation be for any given concentration of all of these subunits. And that is because, in my opinion, limitations of current models. So current models, they lack a molecular basis for the regulators. They don't consider the regulators as inputs. They assume all extruders have identical properties. We're often not modeling binding, unbinding from DNA. We often assume that all of the complexes once on chromatin are active. So far, models are really typically fit to high-C data to understand any perturbation. Okay, and this has been sort of the elephant in the room for me. We had it on the wall in Leonid's lab for a while when I was a PhD student. It's come back to haunt me intermittently. So this is the elephant in the room, a cartoon of a loop extruder. So they've been great. Liobas explained how they can explain chromosome packaging and mitosis, how you can build these backbones out of collisions. However, in this current approach, we're sort of limited to fitting models to high-C data and deciphering the wiggling of the elephant's trunk. Some of you might recognize this elephant. It's from the paper Drawing an Elephant with 4 Complex Parameters. This paper was motivated by an encounter between Freeman Dyson and Enrico Fermi. You can watch Dyson telling the story on YouTube. It's pretty entertaining. I recommend it. Basically, Dyson is very excited. He brings Fermi some predictions from a new model he's been working very hard on with his grad students. Fermi is just unimpressed. And he tells Dyson, with 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk. And ultimately, this Dyson is just totally crestfallen. He abandons this class of models for more fruitful endeavors. So, do we need to abandon loop extrusion? No. So if you bear with me, I'll share how we are able to make the trunk wiggle without the need to change the parameters. And our proposal, in short, is that by lifting some of the limitations of existing models, modeling the chemistry of subunit interchange is a way to build this mapping and not be haunted by these elephants. Okay, so I'll explain our approach to using biophysical data by introducing a 2-state model before addressing the first major limitation with a multi-state model of cohesin. So imagine there's 2 states, an unloaded U state and an active A state. Transitions between these 2 states would be parameterized by 2 unknown rates, K on and K off. You can write down an ODE for the concentrations of the unbound and active states in terms of these rates. How can we figure out these rates? As very nicely introduced by Gordana also. We can use FRAP. So FRAP provides biophysical measurements of the bound fraction in resonance time. The basic idea, right? You have some fluorescent labeled molecules, you bleach an area, you wait, particle positions exchange, fluorescent signal will re-equilibrate. Here's data from Gordana's paper of experimental pre-bleach, post-bleach, recovery, and the curves that can be used to extract lifetime. So we use them. So in HeLa, lifetime appears to be about 800 seconds. The fraction bound appears to be about 65%. We can use these 2 known factors, the 2 equations, to calculate K on and K off. We can obtain the number of extruders per KB in our simulations by using the absolute abundance from mass spec divided by the length of the HeLa genome. Okay, so hopefully everyone's on board with that. What about the other cohesin regulators? So our goal was to obtain the simplest model still able to capture different roles of the regulators WAPL, PDS5, and NIPBL. And the key to this is that our FRAP measurements, again, for each of these regulators. And what interesting thing that these reveal is that the residence time of the core complex, including RAD21, is much longer than the subunits. The core complex has residence time of tens of minutes. In contrast, these regulators are all in this minute time scale. So we have this picture where there's this slow on-off of cohesin and then more rapid exchanges of regulatory proteins on sort of a platform. make progress, we need to make assumptions, right? So here are our current simplifying assumptions. If we make them, we can entirely constrain the model using biophysical data. So first, RAD21 acts as a proxy. The dynamics acts for a proxy of those of the core complex. Regulators require the core complex to bind all chromatin. We neglect states with co-bound regulators, and we assume that for our purposes now, PS5A and B are the same. We don't have CTCF, there's no acetylation, there's no STAG1 and STAG2, etc. So for me, the kind of this a wonderful thing about modeling this way is that at least our assumptions are perfectly clear in a way that might not be from a diagram. Some of these are probably too strong, so I'm happy to discuss which assumptions might be the most interesting to relax and what data might be required in the Q&A. Ask me. Okay, so if we have 3 subunits in the core complex, we have an unbound state and 4 bound states, each representing either the bare core complex, the core complex co-bound with the NIPBL, the core complex co-bound with PDS5, or the core complex co-bound with WAPL. We have FRAP, lifetime, and bound fraction for each of these 4 molecular species. So we have 8 observations that we can constrain a reaction network with up to 8 edges. So some of the simplest such networks are completely reversible, and they have linear topologies shown here, branch topologies, and star topologies. I've drawn all of the reversible, the completely reversible networks, the zoo, where the states are the circles, the colored circles, and the transitions are the red arrows. So we can write down the equations, determine the 8 rates, and actually, interestingly, none of these networks can agree with experimental observations. And specifically, some can fit wild-type parameters, but none of them increase the bound fraction on chromatin after depletion of a subunit, as observed for WAPL. So they all fail. So we need some, if we have only 8 rates, we need some irreversible transitions. So the next set of reaction networks we considered, they're not completely reversible. They have an irreversible transition into and out of the unbound state. What about the ordering or identities of the other states? So we have strong expectations from the years of biochemistry performed on this complex. In the computer, though, we have the luxury, let's just test all of them, right, in an unbiased fashion, see what falls out. So with 4 bound states, there are 24 possible orderings. I'm drawing all of them as a bunch of pentagons with the white unbound state at the top, transitions between them. And for each of these orderings, we can write down the equations, determine the rates from the measurements. What we see is that actually only 8 orderings are able to be parameterized by that data. And then if we additionally require that just qualitatively, that the bound fraction of RAD21 increases with WAPL, increases with PDS5, and decreases with NIPBL, we get one consistent reaction network. There's one cycle under these constraints that agrees at all with experimental data, that even has a chance. And this is it. So I want to understand it in terms of a lattice model for loop extrusion that it specifies. So we start out with this unloaded state. When extruders transition from the unbound state to the bound NIPBL state, proportional to this K on, the free NIPBL and the free RAD21, we load extruders on chromatin. In our model, when they're in the blue state, we are assuming they're moving outwards deterministically. They're reflecting the motor activity of the NIPBL co-bound state. In all the other bound states, they're just not moving. They're barriers, actually, to moving cohesins. And then if we were to leave to the unbound state, dissociating WAPL and RAD21, we remove an extruder from the lattice. Okay, what does this model look like? It spends about 35% of its time unbound, that's from the constraints, only about 17% of the time in the actively extruding NIPBL state. And these are the current assumptions baked into the last model. We're only extruding in NIPBL co-bound, we're not bypassing each other, we're enlarging symmetrically for now, there's no pole2, sister cohesion, or other obstacles. Let's see how far we can get just with these very simple assumptions. So if we want to compare to model predictions to high C, we connect this multi-state 1D lattice model with a 3D model of the polymer as previously. We're also working with folks from Open2C to develop open source software for simulating populars. Happy to chat more about that as well. We generate trajectories, polymer conformations, extract observables that we can compare to high C, and we're very happy. So this plot shows the classic contact frequency versus genomic distance for the simulations in red and experimental data with a dashed line. We have pretty good agreement over about 2 decades genomic distance without fitting to the high C data. This just all these parameters are coming from biophysical measurements. We don't have to throw out loop extrusion yet, unlike Fermi's pseudo-meson scalar theory. So what about perturbations? So perturbations in this model are straightforward. We just input whatever change to the wild type abundances, turn a knob, simulating WAPL RNAi, run the stochastic simulations, extract lifetimes, bound fractions, etc., compare to the wild type values. This plot shows the fold increase for a lifetime as a function of depletion level on the x-axis. First for WAPL, it goes up. And then PDS5 also goes up, not as much. And what we see is in a similar ballpark to the observations reported from FRAP. And this is something that didn't go into the model, so we're pretty excited that we could also explain this. We can explain not only wild-type genome folding patterns, but also depletion, quantitative depletion FRAP observations. We found it useful to also visualize the impact of the different regulators versus multiple extrusion parameters on 2D plots. So this plot shows the lifetime on the x-axis versus the bound fraction for 4 different subunit depletions. They depart from the wild type level, where the dots are 1% increments of depletion from 0 to 99%. So you can see this WAPL, let's focus on that. WAPL is increasing bound fraction a bit, and noticing the log scale on the x-axis is increasing the lifetime a whole lot. And something that comes out of this model is that different subunits really do have different roles for modulating extrusion parameters, just depending upon where they are in the reaction cycle. And actually, and more importantly, for interpreting experimental data, even in this very simple model, right, depletions of a single regulator can often modulate more than one parameter. We also find that the relationship between the depletion levels and the extrusion properties is generally nonlinear. Something else that we can do in the computer is understand why the reaction cycle we looked at is successful. So this is an unsuccessful topology. We explored related topologies that are unsuccessful. Here we call this a NIPBL excursion model, where we start unbound, we load not into NIPBL state, but into a bare RAD21 state. We have a exchange with NIPBL that can happen reversibly as well as traversal through the rest of the cycle. What the issue with this model is that if you look at bound fraction as a function of NIPBL depletion level, it barely changes as a function of NIPBL depletion level, which we know we need to do to agree with experimental data. So this is out. So we can also rule out models that are inconsistent with experimental data using the same kind of reaction topology driven approach. Last, I want to show how we can use some of the same framework to make predictions for 3D conformation, both for our favorite PFS curves as well as for imaging. So here is contact frequency versus distance for different levels of simulated RAD21 depletion, shown as blue to yellow curves. And as depletion increases, this curve loses its characteristic hump or shoulder that we see for extrusion. And what I hope you appreciate is that as the depletion reaches this 99% level, the yellow curves approach that of the experimental data in the dashed black line, which makes sense because this is auxin-induced degron data, which was estimated to have greater than 99% efficiency. And this is the same plot for WAPL depletion. You can see the curve moves in a complete different direction. It shifts rightward. And in this case, the curves actually have a best agreement closer to 85% depletion, which happens to be the level reported as the RNAi efficiency in the experiment. What about vermicelli? So we can just take the conformations, positions of cohesin, blur out cohesin positions in simulations, the same as you might in a microscope, and compare it, at least qualitatively for now, with experimental data, where we see very homogeneous patterns in the wild type simulations and experiments, and very structured vermicelli structures in WAPL depletion. We predict that WAPL and PDS5 depletion would promote vermicelli formation, NIPBL and RAD21 depletion would oppose it. Okay, so to summarize, by building these biophysical models of the loop extrusion process, we can start to bridge these microscopic observations with the genome-wide consequences. And what I hope you can take away is this picture of interphase cohesin as this multi-state extruder. We can make if we take this approach, we can make predictions for high C mutant lifetimes, state occupancies, and microscopy just from wild-type biophysical data. There's a non-trivial mapping between the regulators and extrusion parameters, and it's a quantitative mapping, right? Like the levels of your depletions really matter if you're probing these things experimentally. And we provide molecular rationale for different things like the increased velocity upon PDS5 depletion. Okay, so all of this, I think, speaks to a really interesting emerging principle, which is that cells seem to be changing their abundance of regulators in order to impact the motor dynamics, right? You have these motors everywhere. The cell should make use of them. A really beautiful example from recent literature is in B cells, where they seem to globally downregulate WAPL to boost lifetime of extruders and ensure proper VDJ recombination. So we keep on seeing these new examples of how fundamental chromosome processes are harnessed by cells. My hope is that we can use the models, the type that I've described here, to understand when and how it should happen. So that I'd like to acknowledge Max again and the rest of the folks in the group, as well as our long-term collaborators, Alfej and Rini, and folks from OpenGC. Thank you, and with that, I'd love to take questions.
Thank you. I'd like to start, well, let's go with Leonid first.
Okay. Beautiful, beautiful work, Jeff. Really enjoyed. I watched it over Zoom at Cold Spring Harbor, but now kind of, yeah, very, very much impressed. I remember in the early days of kind of loop extrusion, there were observations that when you deplete cohesin incompletely, the features still remain. If you remember, this works on thymocytes and stuff like that. And people were kind of curious about this, that you kind of for RET21 depletion, it really needs to go very far down for the features to vanish. But for other components, even partial depletions kind of was sufficient to see the phenotypes. Do you see this in these models as well?
So, in short, there's a sort of a bit longer question. So we see a differential impact on depletion of NIPBL and RAD21 on the contact frequency versus distance curve. NIPBL is lowering the velocity as well as lowering the occupancy of extruders. So it is kind of shifting leftwards as well as kind of losing that shoulder. For now, this model does not have CDCF in it, so we haven't been probing kind of the visibility of patterns per se. But the expectation would be that yes, if we put that in, we would also see a differential response of the patterns to the depletion level of those subunits.
Yeah, so I wanted to ask you a question. It's sort of a comment, sort of a question, and it's going to depend on Kumiko straightening me out. But so we like to think of SMC proteins are SMC proteins. So maybe we would want to say that what you're seeing here we could also extrapolate to condensin. But we've got a problem, which is that if we knock out any of the cohesin subunits in our cells, all the other subunits are gone. They all co-degrade. If we do that with condensin, it couldn't care less. All the other subunits stay around. So when you're doing these depletions, are the people who are doing them that you're depending on carefully measuring the levels of all of the other proteins? That's, I guess, a question for Gordana.
So I guess Gordana would be best posed to answer that.
So
maybe I'll call a friend.
One second, I think it's a microphone. Sorry. Here you go.
Thank you. Yeah, so that's true. If you mess up with one subunit, everything changes. So it's for example, even if you mess up with isoforms like STAG1 or STAG2, deplete one, the other one is there, but it will be upregulated. So it's everything somehow connected.
That's messy biology.
Current assumption of the model: no feedback between at least in the short enough time scale of an experimental observation here, that we have not sufficiently disrupted the overall abundance at the protein level of these different factors besides the one being depleted. That's another assumption that was baked in but was not listed. So thank you.
You got to have something to keep you busy for the next 10 years.
Okay, but I just want to continue on the two comments we just heard. I think that's fine the way you're doing. It's actually already a tour de force to try to add all these combinations into the game. But like what you're saying is that even so, the experiments sometimes are linear perturbations. You perturb one protein or another protein, one domain or one thing. The interactions, you may have some comparativity. Your model doesn't have any comparativity. Everything is independent. And I think it's great. Let's see how much can you get away with it. But I think in the long run, you're going to feel that based also the experiments you do by just perturbing single things, these things are actually acting together like we just heard.
Yeah, so maybe I'll just kind of answer the first part of that, or kind of expand on the first part of that, which is there's no cooperativity baked into the model. However, there is this reaction network. So properties of anything downstream of what is depleted are going to change. So in general, you can think of if you deplete something, the levels of everything downstream of it in the network on chromatin will change. And I didn't have time to show it here, but we can also do, of course, pairwise depletions in the simulations and then compare those results with experiments. And what we see is, at least for WAPL-NIPL co-depletion, we're actually doing kind of remarkably well. So to first order, we would argue that the system is kind of linear, even when you're depleting with these auxin-inducible degrons down to 99%. So I think that's actually kind of, from a modeling perspective, is an interesting aspect of the system.
I think it's interesting, but I think it's going to be equally interesting. And it's great that you're getting this sort of, you're not putting into the model direct cooperative, but you get this sort of this combined effect coming together down the road. But it would be interesting also to figure out the case where you fail spectacularly, right? So basically, I think that's going to be equally important to give us some understanding of what's going on.
Yeah, so I was, so speaking of that, so one thing that is interesting, and so I told you about models that we could rule out. One other thing that we can do is potentially rule in different models. Oh, that would be great, yeah.
Could we have the projector back, please?
So we can also maybe use this same approach to rule things in. So I told you we were assuming strict exchange of subunits. There's a proposal in literature which was mentioned, is that maybe instead of sort of PDS5 and WAPL exchanging with PDS5, maybe PDS5 is there, WAPL co-binds, and then everything is dissociated at once. So as you might imagine, this sort of simple topology is going to make a different prediction. Similar predictions for many things, actually. With available measurements, they're not distinguishable. However, if one was able to do the FRAP for the PDS5 subunit after degrading WAPL, that would actually, according to our modeling, distinguish between this strict exchange model and a strict co-binding model. So I think that's a very interesting avenue to explore, and we're starting to do that, and yeah, I think it's going to be very exciting.
Looking forward to it.
No, my computer's, it's okay.
We'd like to see that.
Okay.
Oh, there you are.
Yeah, we're back. So yeah, this is the same reaction cycle, except instead of a exchange here, we're having WAPL coming in on top of the PDS5. And you can see this is the same lifetime as a function of distance. We actually, in this model, slightly overestimate the increase in lifetime. We actually do slightly better in terms of the impact of the bound fraction. Basically, requiring strict co-binding just makes PDS5 depletion behave a little more similarly to WAPL depletion. So that's something that, yeah, I'm hoping to chat more with Gordana with later today or in the next couple of days.
Anybody else? Thank you very much. Really good. Gordana, yeah. Just a second. Here you go. Meet me halfway.
Okay, so this R that you have in orange, is this, would that be just a core cohesin complex?
Yeah.
And is there any evidence that core cohesin complex, like with 4 subunits, I suppose, can stay on chromatin?
If it's not bound by anything?
Yeah.
I'm not sure if there's any direct evidence of that, but if you, so actually we realize you have to have that. It's kind of strictly required because otherwise, if you just multiply the abundances, you get a number that's not enough for the bound fraction of cohesin. So if you multiply if you require that all these other guys are on chromatin when they're bound to rad21, you need this bare state on chromatin as well, just by algebra.
Okay, somehow my feeling was that it's not possible to have it, but it was just a feeling. So.
Well, yeah, so this bare complex is also including stag 1 or 2, which we're not representing here. We're just assuming that it's going with RAD21. Yeah.
Thanks a lot. So our last speaker for this session is one of the organizers, Aleksandra Galitsyna. I hope I said that right. Thank you very much.
Okay, I'm really honored to conclude today's session and today's day on loop extrusion. And here is one more story that we uncovered quite some time ago on the new structures that we call fountains of chromatin. And this was done in Leonid Mirny lab at MIT. And there we got curious about developmental experimental systems where you have an opportunity to look into time series of progressing from just a single cell into the whole organism. And this is in particular zebrafish, where up until 3 hours, the genome of the embryo is silent, and at 3 hours, you have a rapid onset of transcription of the embryo genome. Before that, it exists purely on maternal RNAs, and then at 3 hours, you need rapidly to activate large amounts of genes. And it was interesting for us what happens with the 3D genome organization during this process. And our collaborators from Germany performed experiments before and after ZGA, and after then several time points after ZGA. And they saw that before zygotic genome activation, the genome is not folded. Like, it's not folded into any particular structures. It's just plain diagonal, featureless. And then after ZGA, you got emergence of typical features of chromatin that we also see in regular cells, such as compartments and insulation. And then these features become even more pronounced in further development. However, if you look closer to the diagonal of the embryo's high C, you will see that there are features right after ZGA that do not look as anything that we've known before, not as domains or as stripes. These are also not compartments because they're all on diagonal. And the structures, if you zoom in, they look like something which is perpendicular to the main diagonal of high C, and that doesn't have boundaries. And there are many examples of them in the genome. And if we create the algorithm for the automatic annotation of these structures, we can find around 1,500 of them. And on average, they indeed form this nice structure. We can call it hairpin, but we also prefer the name fountain. So in fact, we found these structures not only in zebrafish, but if we look at the zygotic genome activation of other species, like vertebrates, Medaka, and Xenopus, for some reason it's not displaying. Yeah, here it goes. You can also find the structures in their embryogenesis as well at the onset of zygotic genome activation. And in fact, people from other research groups, they keep reporting the similar architectural features for completely other experimental systems, including small DP thymocytes from Matthias Merkenschlager group. Also, the structures emerge in the double knockouts of CTCF and WOPL. This is the work from the Wutz lab. And finally, the structures are abundant in the 3D genome of lower species such as C. elegans, where there is no CTCF, where there are no boundary elements, but extrusion is present. So we were curious about what the structures are associated with at the level of epigenetics, and we have the nice annotation of fountains in zebrafish genome. So we can try to figure out what are the chromatin factors associated with it, and these are, if we pile all the fountains together, we can see that the active chromatin marks, open chromatin, and pioneering transcription factors are all enriched at the positions of fountains. So this is actually active chromatin, which is here in the center of this fountain structure. But the situation is not so simple. We can try to dig deeper, and there is a nice annotation of chromatin states for zebrafish embryos, where in another group, people took holotoxic peaks and different histone modifications for zebrafish, and they took a HMM model that predicted 10 different chromatin states, including one of them, which is transcription start sites, and we can take a look how much the fountains will be enriched at this particular chromatin state. And then we can look at many other different chromatin states that they annotated, including transcription flanks, enhancers, flanks of enhancers, weak enhancers, poised chromatin, repressed, and quiescent chromatin. So we can see that fountains are somewhat enriched in transcription start sites, for example, but this is not properly controlled because we do not know whether any ataxic peak can also be happening at fountain. And if we shuffle background appropriately, we can actually see that factual enrichment of this chromatin state at fountains is not that different from the shuffled background. And in fact, it's just what we expect. So if we repeat the same for many different chromatin states, we will see that those that are very much enriched over the background will be the enhancers. And it was a first hint for us that something interesting is happening here, because why enhancers would be so much associated with fountains. And we performed more ab initio analysis of Hi-C, where we took multiple genomic bins without knowing which one of them are fountains, or they're just like plain chromatin. And then we took H3K4 monomethylation and trimethylation and plotted them against each other. And we had a nice distribution of all genomic bins. And then we split them into 4 pieces. Those that do not have any of these histone marks, those that have both of them, or either. Meaning that orange set here will be enhancer state, and this will be a promoter state. And then we can nicely see that the fountains will be formed only at those that are enhancers. Even if we have both these marks, fountains will be more overridden by the typical insulation pattern, and promoters will be nicely associated with insulation pattern. So, indeed, we further tried to confirm that there is a strong association of fountains with the enhancers. And in this process of zygotic genome activation, the first factors to open the chromatin are pioneering transcription factors, PO, NANOC, and SOX. And we can deplete all of them. We can create a very complicated experimental systems where the embryos will be starting to develop without these 3 pioneering transcription factors. And in this situation, we can take the regions where indeed the factors disrupted the chromatin openness, the enhancers are no longer there, and this is an example for the enhancer of FOXP2 gene. And indeed, at this location, the transcription factor abolishment leads to absence of any regulatory event in this position of the genome, and at the same time it leads to abolishment of the fountain. So there is indeed linkage between formation of the structures and the presence of transcription factor dependent enhancer. And now, if this was just one example, but if we take the whole genome data and we deplete all the transcription factors and we take a look at those open chromatin peaks that were strong, they were supposed to be important for activation of zebrafish genome, and they went down during depletion of our transcription factors. And it turns out that indeed those were very, very strong fountains. And in knockout, in triple knockout of the transcription factors, the signature will be gone. So the fountains are those ataxic peaks that were bound by the transcription factors, and their abolishment leads to disappearance of the fountains. Okay. Then we hypothesized what might be the mechanism of formation of the structures, because we've learned that no ataxic peak, no fountain, but why at enhancer peak there should be formation of some structure. And just a reminder of mechanism of loop extrusion, that usually the feature that we observe is this nice insulation, that indeed in zebrafish emerges later in development at CTCF boundaries. And first, we tried to look at the fountains versus boundaries, and CTCF was not enriched at fountains. It was enriched at late boundaries only, which means that CTCF is not involved in the formation of these nice structures. Next, we took cohesin, and cohesin indeed was peaking at the late boundaries, because we know that cohesin will be accumulating at the stop points. But also cohesin will be accumulated at the fountains as well. It is a very different type of enrichment though, because late boundaries will have this nice peak, and fountains will be more broadly enriched in cohesin. So just for your information, this is plus minus 200 kb, and this peak will be plus minus 50 kb around the fountain center. We suggested that this might be some interesting mechanism that we observed before. And we saw that in bacteria, where there are SMC-like proteins loading at one particular site, and then cohesin progresses from this loading site on both sides, creating the nice hairpin structure, as we called it back in the days. So it will be secondary diagonal to the high C. However, what makes me very excited is that fountains are not, in fact, the hairpins. They are some different structures that are getting broader with the distance from the diagonal, which suggests that there might be some additional complication involved, and we need somehow to modify this simplistic picture of emergence of the fountains. And indeed, if we introduce just a simple modification of the basic loop extrusion, if we assume that there is a background of cohesins and the cohesins are non-bypassing, then we can pretty fast notice that cohesins loaded will be stuck in interactions with these background cohesins, and it can happen randomly, because each leg will be stuck independently from each other, and this will create the broadening of the fountain. And just to show it a little bit more in terms of how it will look like in simulations, imagine that there is cohesin that has loaded at this site, and then it progresses symmetrically with both legs, but then it encounters some barrier only on one side that causes the deflection of the track of this cohesin, and then it will progress maybe after this boundary is gone. The cohesin will progress again perpendicular to the main diagonal, symmetric, but multiple events like that, which can happen randomly at different positions, on average, will lead to this more broad structure that we can now call fountain, and we simulate, and simulations nicely agree with this picture of more broad structures than just hairpins. Okay. What about confirmation that these structures are indeed cohesin dependent? It is not possible to create cohesin mutant in zebrafish, because embryos won't survive. However, we can try to take a look at other experimental systems that are available. And for example, these are mouse embryonic stem cells. And there is a nice data with different degradations of multiple factors. And this is the alignment of different acetylation plus enhancer mark sites. And you can see that on average, the structures are forming fountain-like structures. And then in red 21 degradation, the structures are gone. So this is already a hint for us that if there are indeed fountains in mouse, on mouse enhancers, they would be also cohesin sensitive. Okay, we can also take CTCF degradation, and in this case, the fountain will become, they become weaker, but they also become more perpendicular to the diagonal and more prolonged. Which is very much in line with our model of random barriers to the sides of cohesin loading, because in this case, there were boundaries, maybe other CTCFs or maybe other cohesins on the sides. And now there are no CTCFs, and the cohesin can progress further from the main diagonal to create this fountain structure. And finally, if we have wobble, then the fountain structure will be more pronounced and large as expected because we don't have cohesin unloading that frequent. So the really fascinating question for me is what is the function behind fountain signature? And here I would like to just discuss some of interpretations that I really, really like. And one of them is that maybe enhancers need some kind of support to reach out to their promoters at the onset of genome folding, when the embryo needs to develop and activate many genes, and maybe the fountains can help to facilitate the interactions of the enhancer with the promoter. And this is in a scenario where multiple cohesins were loaded at the same platform of enhancer, and because they are stuck at this position, it may cause additional interaction with the promoter. Alternatively, it might be that enhancers that we see in zebrafish are not in fact simple enhancers. They might be enhancers of another enhancers in the surrounding areas. And this is an example of cohesin loading at some locus and then progressing up until it interacts, up until it encounters simple enhancer, and now the gene will be zipped together with this enhancer. And finally, alternative model that might work here is that is coming from the observation that, in fact, when cohesin is loading at single site, it is likely to isolate a little bit this region where it was loaded from the surrounding areas. We can see that already from the pileup plots that you have seen, that the enhancer itself is forming white stripes to the side, which means that it's not really enriched in interactions with the surroundings. So maybe it happens because the cells would like to isolate the active element from the repressive environment of the surroundings. And it also makes sense because zebrafish embryos are usually, like, before the onset of transcription, the chromatin is inactive, and we need to rapidly activate a small subset of genes, and maybe that is the process that helps to maintain the status of these regions active. Okay, and finally, another interesting observation that I would like to highlight here is that we know that embryos are also very rapidly dividing. And in zebrafish, the division happens every 15 minutes, so it's like very, very fast. And maybe it's somehow formation of the structures is interconnected with the cell cycle. And here we can try to look at some other experimental system, and this is from Gerrit Blobel lab, the data on mouse erythroblastoid cell line, where they synchronized the cells, and they performed Hi-C on that, and it is very good quality Hi-C. So you can see that at prometaphase there are no structures formed. So, and if we take enhancers, then we see that there is also no enrichment and practically no structures there. But then, as early as at anatelophase, you can see that there is formation of some structures, and the contrast here is not enough to see that on the slides, but if you do the pileup, you can already see this emergence of the structures around enhancers. And it becomes more and more pronounced with the progression into G1. And now you can see there are several structures like that. So this is a characteristic signature of the fountain, where there is this halo of interactions which looks like perpendicular to the main diagonal, plus this white depletions of the interactions to the side of this region. And there is another structure here, which is a little bit tilted to the side. You can see that it's like forming this structure to the left. So it's kind of the fountain which is tilted to the left side. And yeah, they become even more stronger in the progression into G1. With that, I would like to conclude with these structures, fountains, that are first to form in the development, and it looks like they're enhancer-specific, at least in zebrafish, and they depend on pioneering factors binding. We assume that it's targeted cohesin loading at enhancers, and feel free to check out our bioarchive. With that, I would like to conclude and acknowledge all the collaborators involved, and particularly Daria Onishuk at University of Freiburg, Sergei Ulyanov, Mikhail Gelfand, and their bioinformatics team, and me in the lab, of course, in all the people, including Open2C community that I can't stop acknowledging because they're doing really great job on high-C analysis. And yeah, thank you, organizers, and for such an amazing conference, and thank you all for listening. Yeah.
Thank you very much. It's really exciting. Yamini, can you do that side? Can you take first to Magda over?
Yeah.
Beautiful work. I just wonder whether there is something, some feature of this sequence that is particularly pronounced at these enhancers that form these fountains. Are they very GC rich or?
Yeah, that's interesting. I will just bring up the picture of a typical fountain here, just for illustration. Yeah, here, for example. So we tried the approach with a neural network where you can try to extract the fountain signature and then try to see what DNA motif was associated with it. And it looks like these are different pieces of transcription factors bounded enhancers, so this pioneering transcription factors, and you will see all sorts of combinations of the motifs of these transcription factors at the positions of the fountains, which is another confirmation that, indeed they're happening at enhancers, and you see enhancer motifs enriched at the fountains. Other than that, we do not see any particularly enriched sequences there, like no particular GC content, nothing else so interesting.
Thank you.
Hi. Very nice talk, thank you. I wanted to ask again about WAPL depletion. So in the case of WAPL depletion, we believe we find more cohesin on chromatin. So that means that the loading would be more than in the case of just unperturbed cells. So do you see more fountains?
We see, that's a good question. We do not see, I'm not sure we can detect them ab initio in mouse cells because they are too much obscured by CTCF sites. That's why I need to do very complicated pile-ups, cleaned up of CTCF signature for that. And what we can see at this signature is that the fountains become more prolonged and more wide, which is in agreement with the fact that there are more encounters with the background, and maybe that might be in line with your notion that there is more cohesin at the chromatin. So there is also more background.
Okay, thank you. So you can extrapolate actually TAD boundaries from fountains, and can that be used for HeLa cells, for example?
It is a very interesting notion. I cannot find the fountain signature for cancer cell lines in humans for some reason, not in HeLa and not in HCT116, for example. This is a mystery why. Maybe some cells just do not load cohesin on their enhancers, and it's more uniform loading throughout the genome. So it's unclear, but not all cells have this nice signature.
Thank you.
Here. Very nice. So I'm wondering about the cohesin loading mechanism. So do you think it's because pioneer factors open chromatin that it allows for the loading of cohesin there, or is there some more specific protein-protein interactions between cohesin and those factors?
I agree, and I think it's a very hard question to untangle because we know that cohesin has the preference to some open DNA, which is not bound by nucleosomes. But can it be something more specific? For mouse, we know that some subunits of cohesin might be interacting with pioneering factors. We know that from like experimental techniques where we do mass spectrometry on the cross-linked cross-linked proteins. But this is some thing that is really hard to do on zebrafish and on this large scale. So I do not think we currently can answer whether it's something very specific to some particular protein that loads at enhancers and then recruits cohesin, or it's the property of cohesin to be just loaded at plausible open chromatin sites. And yeah, if you have ideas how to disentangle it, I will be very happy to chat.
Okay, this was very nice, and I think but I have a couple questions. I looked at the data, and I think I really enjoy your talk, and I think but how much maybe these fountains are, just two questions, they may be like a dynamic effect. This is not like a structural effect. These things are just moving around, forming and expanding, and you're trying to give a structure for it, but maybe it's just something dynamic effect coming. And the other thing is, I agree with you that in order to have the fountain, you have to have some sort of a multi-cohesion interactions. You have the idea, you have this collision model, but you could come up with some other explanations of having multi-collision interactions, basically, maybe some traffic jam on the connection, other things like that. How can we disentangle that?
I really like your first point on the dynamics. So I think that there might be an interesting interplay of fountains with the dynamical regulation of the nuclei at this development point. So yeah, thank you for that notion. And it's something that might be worth investigating further in terms of more theoretical models on top of that. But the second question you raise appropriately, because it can be some other type of barriers in the genome. It might be some random CTCFs or, I don't know, some other random obstacles in the vicinity. And for that, we just use the minimal possible model here. But yeah, indeed, we try to explore them with simulations. And indeed, other alternative models can explain that as well. It's just like the terms of what is the easiest model to explain it. Yeah, but we provide different alternatives with our work, I guess. Yeah. Nice, thank you.
Nice talk, Sasha. I was curious, I think I might have missed it, but did you say the fold enrichment that you needed, say for mouse, in loading at the enhancers in order to
that is a very interesting aspect. Yeah, I made the details of the simulations here, but it looks like just 10 times more enrichment at the enhancers is enough to generate nice fountain signature, which is best correlated with the fountains in zebrafish. So it's just 10-fold increase. It's not something like 100 or 1,000 fold increase. Something not that large.
And so the question with that was, does this fold increase needed to recapitulate the high C also give you the correct chip-seq enrichment? Because would you see that or not?
Yeah. I don't think I have slides on that, but it's very beautiful to see this volcano at the simulated chip-seq profile. Here I have it for the average data, but it's nicely reproduced in the simulations. This one. So where I show this volcano on the cohesin signature, it is reproducible in the simulations as well, because cohesins are loading at the initial site here, but then they are stuck in the other surrounding cohesin, which creates this traffic jam for cohesins, and that's what creates this profile of in silico chip-seq coverage. Interestingly, in simulations, we have a dip in the middle because cohesin that has loaded immediately runs away from the site. And that's what we cannot see in the data because we cannot pinpoint precisely the position where cohesin loads and average around it. Yeah.
Hey, beautiful work. I can't help but notice, and sorry if this is a sort of a stupid biology agnostic theorist's question, in this session of the conference we've had 2 talks on features where cohesin seems to be loaded more densely than in the rest of the chromosome, and you have these sort of depletion stripes that cross out into the rest of the chromosome, so these S compartments and the fountains. Do you think it's possible that there's some connection between the two? So for example, if in the simulation you were to place strong extrusion boundaries at the sides of the fountains, do you get something that looks like one of these S compartments?
It is an interesting connection indeed. Fountains do not checker, so they do not create compartments with other fountains in the genome, which makes it really similar to S compartment. I agree, but it is much more narrow than NS compartments. Maybe some micro-C experiments will shed light on the intrinsic structures of the fountains, and maybe we will see something like, I don't know, something like little S compartment-like structure, and we can untangle it more. But similarity, like superficial similarity, is there. They do not checkerboard. They presumably load cohesin, and I appreciate this note. It's nice.
That was really beautiful, Aleksandra, and I understood your talk because I attended your summer workshop on the first day. So really beautiful.
Easier.
But I really like this cool concept you introduced of the enhancer of enhancers towards the end. So enhancer looping is thought to rely on transcription of the enhancer, right? These little RNAs that come out of the enhancer, the eRNAs. Have you considered poisoning the system with like acetylcholine, or like just briefly for 20 minutes blocking transcription? By RNA pol 1, 2, or 3 to see if it changes the way these things form, these little fountains form? Thanks.
I don't think that experiment has been done, so we do not have an answer. But yeah, it's an interesting and nice question. So if we respond so much to the abolishment of enhancer, maybe transcription is also playing a role, and maybe there is some interchange between enhancer and promoter, which is important here, and it might be transcription.
It could also just be supercoiling, right? It could also just be supercoiling, driven by transcription.
In C. elegans, the colleagues who are co-submitting the work with us, they tried that. So they tried to remove supercoiling, and they see that the fountain structures are still there. And they have done that in C. elegans, which is of course not entirely our experimental system, but still we are confident that it's not supercoiling and less dependent on transcription. Yeah.
Thank you, Yemeni. So thank you very much, Aleksandra, and thank all the speakers. Let's do that quickly.